Hi, guys. Welcome, everybody, to the grand finale, night 5 of AP Biocrams. Whew. I am so happy you guys are here. We have a fun-filled night. We're going to get through all the content. We're going to do practice multiple choice, practice FRQ, math problems, and we're going to do it all in 3 hours, okay? So I am so excited that you guys are here with us. Please make sure that you're following us on @ThinkFiveable on our social. We have lots of fun videos, tips and tricks, and unit guides for you guys. You're going to find lots of resources, so please make sure that you follow us. In terms of who's going to be with you, I am one of the teacher hosts. My name is Sabrina Shumate. I am in Virginia Beach, Virginia. I teach be sharing tonight with another teacher host, and her name is Kari, and she's going to be taking over the second half with you guys. We have two amazing student hosts with us tonight. We have Rashi and we have Jeffrey. So Rashi, there's Rashi, and Jeffrey, there he is. They're going to be with you tonight. And so Rashi and Jeffrey, if you could, let's have Rashi go first. Could you tell everybody your name, where you are presenting from, and then if you had a time machine, if you would rather be able to go backwards in time, or would you rather be able to go forwards in time?
Hi everyone, I'm Rashi Lakutia and I'm presenting from Tampa, Florida. And if I had a time machine, I'd want to go forward in time because I feel like I already know what happened in the past. So there's not much I could do. But going forward in time, there's so much more you can explore and learn about the world that it'd just be so much more interesting.
Hey everyone, I'm Jeffrey. I'm from Minnesota. If I had a time machine, I'd rather go in the past to see the Protestant Reformation.
Very nice. All right, my loves. Thank you so much. If you guys, what about you guys in the chat? If you had a time machine, would you rather go in the past or would you rather go into the future? And you can go as far into the past as you want or as far into the future as you want. What would you guys prefer? And nothing would happen to you. So like if you want to visit the dinosaurs, you could visit the dinosaurs and they wouldn't like chomp you up. Pass. I would fix some things. Pass. Some of you guys would go back and retake your AP test. That's kind of funny. The future to get you guys are so funny. So I can see you're all centered on this test. Understood. All right, we're gonna go right into what we're gonna do today. All right, so you hopefully have completed 4 nights of crayons. If you have not done 4 nights with us in a row, you have access to all the videos. Since you guys are here, you can go back into the lobby and see all the videos and replay them. And if you haven't attended a single one and tonight's your first one, big deep breath, no worries. We are going to be reviewing every single thing. So you are going to be in good shape. I am so appreciative that you guys are choosing to spend the night before your AP test with us. We're going to help you. I'm on the East Coast, so I will be with my East Coast people from 7 to 10 p.m. And everyone else, you guys will have a little more time to study than the rest of us. So today is going to be focused purely on reviewing everything. Okay, so we're just going to kind of bear down and go with it. In terms of what you need to have with you, you want to have some snacks. You always have your snackies. You have to have water. I would have a calculator, something to write with and something to write on. And that would be kind of all the things that I would have. So while you guys kind of get that settled, the best way to make the most out of the cram is going to be to take notes. So don't just listen to me. You want to write things down so that you have it. I'll kind of give you tips and tricks and things to write down. I'll let you know what to write down. We have a cram hub in Fiveable that has everything that you can want. There's even this beautiful 1-pager cheat sheet that I would look at tomorrow morning, like right before you take the test. We have the group chat. If the group chat seems to be a little distracting, then just focus on me and ignore the group chat. No one has to go into the group chat. We do ask if you and to encourage you to ask questions. The student hosts are going to be there replying to your responses. And there's even like a question and answer tab as well. And then we also have some polls. So like if you look on the right-hand side, you'll see like a little thing of polls, and we'll be presenting those throughout the night for you to answer them. So before we get started, we're going to ask you a couple things, a couple of community norms. Number 1, let's please be supportive. We are all nervous. Tomorrow is a big day, but hopefully by the end of our session tonight, you're going to be feeling a lot more comfortable, a lot more confident, and you're going to just totally rock this exam. You're going to crush it, okay? please be patient. We know technology things happen. We know all of a sudden my screen might go blank, but we are going to just keep on moving, okay? And please be kind. That's kind of like a life lesson, really, is to always be kind. We're all real people. I'm an AP Bio teacher, and the students are the students, and we're all going to just try our best, okay? So if you want to win some free money, then please share your cram experience with us on TikTok or Instagram. Here's everything that you need to know, and they will DM you if you're actually going to win, and you can see some of the old videos that we have. All right, so before we get started, I want to do a little check-in. Where are you? What are you feeling at this very moment? Are you a 1? Are some of you a 1? Are some of you a 2? Are some of you a 4? Are you a 9? Are you wishing you just had some pizza? Oh, I got a couple 8s in there. You poor things. A solid 6. Okay, I got 110. Okay. Aw, 7. Denial. That's okay. By the time you're done with us, you're going to know a lot. We got this. Probably a 6. All right, you guys. We're going to have little built-in breaks so we can have little stretches. Just stay focused. We can do this, okay? We're here for you. All right. So throughout the actual session, we're going to ask you kind of just like get to know you questions. So the first question we have is, how many siblings do you have? Are you an only child? Do you have 2 siblings, 3 siblings? 1 too many. Yesterday, we asked a question of what pets do you have, and everyone said a brother or a sister. Kind of funny. Not really kind, but kind of funny. I'm an only child, but I have 2. I have a son and a daughter. Oh, very nice. We got a 3, only child. Only child, baby. Let's hold strong to that. All right, very good. Thank you for sharing. So in terms of the test and what you're going to see, you have 2 parts to the test. You're going to have a multiple choice, which will be 90 minutes. And then you're going to have a 10-minute break. And then you're going to have 90 minutes for your FRQ. Okay. And so in terms of the content, this is kind of showing you the percentages of the test. So the majority of your test is going to be genetics, evolution, and ecology in terms of content. But last night, I was explaining to the students that were with me that you can answer most of the tests without pulling in a whole bunch of detailed biocurriculum. If you have a model or a graph or a data table, you'll be able to answer most of the questions, okay? So I want you to take big, deep breaths and know that we're going to be walking you through this whole event, okay? So in terms of the layout, I just said you have 60 multiple choice, and then you're going to have the 6 FRQs. You're going to have 2 long and then 4 short. And long and short do not refer to how much you were writing. It refers to how many parts. So the first 2 long FRQs, the first one's going to be a data table or graph, and then it's going to be pure analysis questions, okay? It might be part A, B, C, D, E, and then usually the last one's going to be something about content, okay? Then the next one's going to be where you have to make the graph. And then for the short FRQs, maybe you only have a model, a data table or picture, and maybe part A and B. And so I want you to think about your test as an Easter egg hunt, okay? Your job is to just get as many eggs as you can. It doesn't matter the order that you get them or the sequence that you get them. It doesn't matter that you want to do FRQ 1 and then you want to do 4 and then 5. You can do it out of order. It doesn't matter. The point is to get as many as you can, okay? There's no penalty for a wrong answer. So when in doubt for your multiple choice, if you run out of time, just throw it in there, okay? Just randomly guess. Number 2, for your FRQ, you can get part of a section right and then part of it wrong. So like, for example, for a graph, maybe you have A is worth 2 points. You can get 1 point and not the other. So you want to make sure that you do all the ones that you know for sure, and then just go back and try to do the ones that you maybe don't know, okay? So what I'm going to do is I'm going to kind of go through units 1 to 4. So that's basically chemistry and cells, kind of like the easier stuff. And then we're going to kind of go right into some practice questions with that. Okay. So I'm going to kind of ask you guys some questions. So the first part of chemistry talks about water. Water being, H2O, and we know this. Water has unequal charges. And so what was the term that's defined as unequal charges. Starts with a P. What do we say? What is that term that pertains to water? Water is a what molecule? Starts with a P. It means unequal charges in the negative end. What is water? Beautiful. Water is a polar molecule, okay? So if you get an F or Q, anything that talks about water, I want you to talk, if it talks about some sort of special property or you just see water in it, I want you to always say that water is a polar molecule that has unequal charges. It has a positive end and a negative end, so it acts like a magnet. And because it acts like a magnet, that is the basic foundation of all the different properties of water, okay? So always come back to the fact that water is a polar molecule that has a positive end and negative end, and that is going to act like a magnet. And that kind of allows for all the different unique properties of water. So what did we call that property of water where water sticks to other water molecules? What was that called? Is that cohesion or adhesion? Water sticking to water, like it forms the bubbles that we get and surface tension. Cohesion, very good, you guys. Okay, so cohesion is water sticking to water. Now, what I want you to know is when I say water sticking to water, what is the name of the bond that's formed between water molecules? Very good, you guys. Look at you. Hydrogen bonds. Great job. So hydrogen bonds are found between two water molecules, okay? You have the positive hydrogen being attracted to the negative oxygen of the other water molecule. Beautiful, okay? Because it is charged, water can now stick to other charged things. What is that word we use to describe water sticking to other charged things? That's not water. Adhesion, very good, okay? So water sticking to water through hydrogen bonding is cohesion, and water's ability to stick to other things is called adhesion. You guys are rock stars. See, you got this. All right, Rashi, if you could, go ahead and see how they do with this first question. So Rashi, can you read this out loud for them? And then Jeffrey, if you can go ahead and open the poll and let's see if they can get the right answer.
As ice melts, A, water molecules become less tightly packed, B, the water becomes less dense, or C, water molecules are closer together. And the answer is C, water molecules are closer together. It seems like over 70% of people got this right, and then 30%, 70% originally chose A, and 30% chose C. So I guess we should go over this one a little more.
Okay, so I'm even looking at this because as ice melts, the water molecules get spread out and they actually move farther apart from each other. So I think that this was actually going to not be C. So I think that we might have had a typo in this particular answer. So let me explain what happens when ice melts. Okay, so let me take this back. Sorry, you guys, I'm having a moment. When ice melts, not when ice forms. Look, even I got tricked up. Okay, so when ice is formed, it's going to be essentially like packed, like separated apart like this, and it forms these like lattice structures. Okay, and so it kind of freezes and then it kind of spreads out, right? And then as the ice melts, it's going to kind of come closer together, and then it kind of just forms into the liquid. So the answer is actually C. And I think that the majority of us, including myself, actually was going with A because we thought it meant as ice forms, but it was as the ice melts. Okay, so think about it like as the ice melts, so you have these water molecules kind of all going together, and then as it freezes, you form these lattice structures between them, and it kind of creates these holes, and that's what allows the ice to actually float because it has these holes in it. But then as it melts, those spaces kind of go away and the molecules come closer together. So the majority of us, I even misread it with you. But the correct answer is C. Good job, you guys. Very good. All right, so then we went into macromolecules. Okay, so, oh, I know everyone's favorite, macromolecules. Now I'm going to ask you guys some questions, okay? So the 4 macromolecules we had were carbs, lipids, proteins, and then nucleic acids. Between the 4 of these, which one is made up of amino acids? Which one of these are made up of amino acids? Beautiful. Proteins. Proteins are made up of amino acids. Very good. Amino acids are going to be held together by peptide bonds. When you have a whole bunch of those peptide bonds together, we call that a polypeptide. So now we have carbs, lipids, proteins, nucleic acids. Which one of these is going to all be nonpolar? They're all nonpolar. Which one of these is all nonpolar? Lipids, very good. Lipids are all nonpolar. That is a unique property of all lipids, okay? They're all nonpolar. So in terms of lipids, I want you to remember that they're all nonpolar. They also stack on top of each other. And we can have saturated fats and we can have unsaturated fats. Between the 2 of those, sat fat and unsat fat, which ones contain single bonds and only single bonds? Sat or unsat? Which one contains only single bonds? SSS, very good. Sat fats are going to be single bonded, okay, and solid at room temperature. That's how you remember that one, SSS, okay? Unsaturated fats are going to have a double bond, and that's going to make it kind of bend a little bit. So they're not able to stack so easily. They kind of slide over each other, and they're going to stay liquid, and those are going to be your oils, okay? Very good. All right. Now, in terms of proteins, what you have to know is kind of how proteins are going to form their final shape, okay? And so it kind of occurs in 4 steps. We have primary structure, secondary structure, tertiary, and quaternary structure. So primary structure is just the formation of the protein on the ribosome itself, okay? It's a singular sequence. We know the order is very important. It's held together by peptide bonds. Thinking about genetics, what was the term we used to describe the making of the protein on the ribosome? Was it translation or transcription? Which one of those words describes the actual making of the protein on the ribosome? Ooh, there it is. Okay. So translation, very good. So transcription is us making a copy of what we need, and translation is the actual building, that second step. Okay. So it's transcription and then translation. So translation is the building of the actual protein itself on the ribosome. Now, ribosomes can be bound to an organelle or they can be free floating. If the ribosomes are bound to an organelle, what is the name of the organelle the ribosomes are bound to? What is the organelle that ribosomes are bound to? There it is, the ER. Very good. Okay, so we call it the rough ER because the ribosomes are going to be bound to it. So if we are making a protein that's bound to an ER, then it's bound to leave the cell. Okay. If we're making a protein that's attached to a ribosome, then that's the kind of free floating that's going to stay inside the cell. So what you have to know are the type of bonds that are each of these different stages, right? So in primary, we just have peptide bonds. In secondary, we're going to have some hydrogen bonds form. And depending on what the end protein looks like, you're either going to form the alpha helix or the beta pleated sheet, okay? So if your final protein needs to be super flexible, right, like your hair is super flexible, then it's going to form this alpha helix shape. And so you have some hydrogen bonding that's going to form. If it has to be super sturdy, like a hemoglobin, like your red blood cell, then it's going to be a beta pleated sheet, okay? Then the whole molecule folds in on itself in the tertiary step. So in the tertiary step, I want you to remember that this is the first time that we get a whole molecule folding in on itself. And what's folding in and attracting are the R groups that are found in the amino acids. So amino acids all have an amino group, an acid group, a central carbon, and then this R group. And some R groups are positive, some are negative, some have sulfohydryls, okay? Some are hydrophobic, some are hydrophilic. So this is where we start to get the actual shape of the protein itself. And then the very last stage is when we have multiple different proteins coming together. So like to form a red blood cell, it's actually 4 different polypeptide chains that kind of pop in together to form that final shape. Okay, so just make sure you kind of have an idea of the structures and the formations of them. Here is a nice summary chart of everything macromolecules. Okay, you just have to know big picture things. So I want you to know that the monomer of a carb is a monosaccharide, but just look at the word. Mono means one, saccharide means sugar. So it's a little baby sugar, okay? What do we call it if it has 2 sugars? Does anybody remember what 2 sugars is called? What's 2 sugars? If 1 sugar is monosaccharide, what's 2 sugars mean? Disaccharide, very good. Here's my bonus question. Can anybody give me an example of a disaccharide? There was 3 that your teacher probably taught you. What is one example of a disaccharide? Very good. Lactose, sucrose, and maltose. Good job, you guys. Those are disaccharides. Nailed it. Great job. All right, Rashi, if we could go ahead and try the next concept check, and then Jeffrey, if we can go ahead and present the poll for concept check number 2.
A macromolecule has several chains containing carbon rings with 1 oxygen per ring. Which macromolecule must it be? A, lipid, B, nucleic acid, C, carbohydrate, or D, protein?
Not sure what happened to the poll. It doesn't seem like there's. Oh, never mind. There you go. Thank you.
It seems like over 90% of people are saying C, carbohydrate, and that is in fact correct. Good job, guys.
All right, my loves. Great job, you guys. Carbohydrates. Very good. All right, so we're going to move forward through what we know about cells, okay? In terms of the organelles, you just have to really know functions, like nothing that is extreme or difficult. So like we know vacuoles are going to store water. The main structure that we really focus on is the cell membrane. That's kind of like the one that we kind of see over and over and over. And all the other ones, the nucleus, obviously, okay? So what I want you to know about cells is that cells want to be a very specific size. They want to maximize surface area while also kind of keeping an eye on that volume, okay? A cell cannot be too big because it would take too long for things to diffuse in and out of the cell. It cannot be too small because it has to hold all those organelles. So there's kind of this sweet spot. And so what we can do is we can calculate the surface area to volume ratio. You will be given a formula sheet on test day. You do not have to bring it yourself, but you do have to bring a College Board approved calculator. So make sure you go to College Board, check calculator policy, make sure it's approved. Make sure you bring extra batteries. You want to eliminate as many stressors as possible. So I would have my batteries and then I would have my calculator like ready to go. You also want to bring 2 pencils, right, for the multiple choice part, and then you want to bring 2 blue or black pens. So you want to kind of gather that stuff tonight and put it by the door. So that, or you know what, put it in the car, so you don't got to worry about it. But the formula sheet will be given, so you don't have to memorize formulas, which is kind of nice. So they'll give you surface area formulas. They'll give you volume formulas of like a sphere or a cube. And if you're given something in this situation, then they're going to say calculate the surface area to volume ratio. Literally divide it. Find the surface area, divide it by the volume, and then you're going to get a number of some kind. And what cells want to have is they want to have a high surface area to volume ratio. And so the way that they establish surface area to volume ratio, a higher surface area to volume ratio, is by increasing their surface area, okay? So if you think about the folds within a mitochondria, there's lots and lots of folds. If you think about a neuron, which is going to be a cell in your brain, it is super, super, super branchy. Okay. And so they kind of increase their surface area so that they can allow for an increase in efficiency of nutrients, diffusion, being able to respond and to any need that they might have. And so what I want to kind of go into now is the cell membrane. We know the cell membrane is semi-permeable. We know that only certain things can go in and out of the cell. We know that it is going to have a hydrophilic part, which is going to be the heads of those phospholipids. And we know that the tails are hydrophobic, and they all kind of face in with one another. So anything that is going to be hydrophobic, so nonpolar, can easily go through. Anything that's not, so anything that's polar is not going to be able to. So it's going to need a little help, okay? And so we have special proteins that are going to help things get in of the cell membrane when needed. So there are two types of transport. We have active transport and passive transport. Which transport requires energy? Do you guys remember? Active or passive? Which one requires energy? Active, very good. Okay, so active transport requires energy. There are three types of active transport you need to know. Endocytosis, exocytosis. Which one is going to be bringing things into the cell? Is it endo or exo if I'm bringing it into the cell? Endo, very good, okay? Exo are going to be things that I'm going to kind of push out of the cell. Sodium potassium pump is when I just need to create a charge potential of some kind. Passive transport doesn't require any energy. And in nature, molecules like to go from areas of high concentration to low concentration, okay? So I want you to think about concentration and elevation kind of being the same if you get confused between the two. If we are on top of a mountain and I ask you to go down the mountain, you could easily go down the mountain, right? You would just walk right through, gravity pull you down, nice and easy. But if I ask you to go up a mountain, that's going to require a lot more energy. So passive transport is going from areas of high concentration to low concentration. That's going to apply to molecules and also to water. And what was the diffusion of water called? Does anybody remember that? What's the diffusion of water called? Osmosis, very good. Okay, so osmosis is diffusion of water. Good job. Diffusion is simply the movement of molecules from high concentration to low concentration. And facilitated diffusion is when we're going to use proteins to help us kind of push things in. Maybe they're just a little too big. And so we just are going to use these kind of proteins to push things in. That's facilitated diffusion. So it's regular diffusion, but with help. So we call it facilitated. And so what I want to go over is going to be kind of the three different types of tonicities. So we have hypertonic, hypotonic, and isotonic. So my question for you is, of those three tonicities, which one would be occurring if we have water rushing into the cell? Is it hyper, hypo, or iso? If water is rushing into the cell. Hypo, beautiful. When I think of hypo, I think of a cute little hippo, like a hippo is swollen, like hypo is swollen. So water is rushing into the cell, okay? So hyper, iso, hypo. Which one is going to be a cell that's going to be kind of at equilibrium? Is it hyper, hypo, or iso? Iso, very good. So when you have a cell in an isotonic state, a lot of people think that there's going to be no movement, but that's not the case. That's a misconception. I want you to know that cells that are in an isotonic state are going to have no net movement. So you have equal amount of water going in and out, and they're kind of just always at this balance. So that's isotonic. So as a plant cell, or as an animal cell, we want to be in an isotonic state. Okay, that's kind of like our favorite. We don't want to be in hypertonic because we'll shrivel up and die. We don't want to be in hypotonic because we'll explode and die. Those animal cells want to be in the isotonic state. If you are a plant cell, what state do you think they prefer? As a plant cell, which state would you think they prefer? Hyper, hypo, or iso? Hypo, very good. They want to be turgid, right? Plants can't just drink water like we can. they want to be able to be filled with water. So plants prefer that hypotonic state, whereas we prefer the isotonic. What organelle prevents the plant cell from exploding? Like we explode, but why don't plant cells explode? What do plants have that are specific for plants that keep their shape and structure? A cell wall. Very good. So that cell wall keeps their shape and structure for them so that they don't explode. Great job, you guys. All right, we are moving right along into enzymes. Enzymes are catalysts that speed up chemical reactions. We know that they do that by lowering the activation energy. Enzymes are very substrate specific, so they're not going to bind to anything, okay? Enzymes can be reused, and enzymes can build things up and break things down. So there are two vocab words on here, catabolism and anabolism, or catabolic and anabolic. Of those two words, catabolic and anabolic, which one means to break things down? Catabolic, anabolic. Which one means to break things down? Catabolic. Good job, you guys. It's catabolic. Uh-oh, I saw one anabolic. Catabolic, okay? I want you to think about a very cute breakdancing cat, okay? It is breaking things down. It's a breakdancing cat. Catabolic means breaking things down. So when an enzyme is breaking something down, that is a catabolic process. So that would be like digestion, right? And it has to be in the presence of water, right? So water is needed to break things down. Anabolic is when we want to build things up. So when I want to build a protein, then I need to remove water, and that's going to go ahead and build things up. So in my head, I just remember, yes, cats do destroy everything. I agree with that. I think about like a female construction builder, like Anna the builder and the breakdancing cat. Or if you do know that cats do literally break down everything. The other thing I want you to know about enzymes is that they can denature. Okay, so denature basically means you have taken an enzyme out of its optimal conditions and it's starting to impact those bonds that hold that protein together. So when you disrupt those bonds, everything just kind of falls apart like to mush. Okay, so the one misconception that happens a lot, and I want to make sure that you guys are aware of this, is that when it comes to enzymes, if you are given something about what environmental factors can influence an enzyme's rate of activity, what would you say as an environmental factor that could influence rate? What would you say? What are some environmental factors that could influence the rate of an enzyme? Temperature, pH. Beautiful. Very good. Okay, those are kind of the go-to. Salinity for sure. Absolutely. Okay. Those would be your go-tos. Okay. We know that enzymes have very specific ranges that they prefer. But what will happen is that you guys will say enzymes don't like it when they are in a low or too high a pH, or too low or too high of a temperature. Number one, don't use the word like because they're not actually like real things, right? We would say they prefer or they work more efficiently at optimal temperatures. And remember that every enzyme has a very specific range that they prefer. There are enzymes in your stomach that like that lower pH. There are enzymes in your mouth that prefer the higher pH. There are enzymes that prefer really warm temperatures versus really cold. So it's not a very specific temperature. I just want you to make sure that you say that enzymes have specific ranges that are optimal for them. And then when you're out of their optimal ranges, then they're going to denature. Denature means that the bonds are breaking and then essentially they're no longer functioning, but they can reform. Taq polymerase, yes, very good. Nice bringing that back. I like that from our biotech unit. Taq polymerase is the enzyme that likes that really, really high temperature. Very good. All right. Now, enzymes is not going to always want to be created, breaking things down and building things out, right? So we want to be able to turn enzymes on and off based off our availability, right? Like if I am not drinking milk, I probably don't need the enzyme that's going to break down milk. Okay. And so essentially, we have a way of turning enzymes off, and there's two ways. There is competitive inhibition and there's non-competitive inhibition. Okay. Now, between the two, the C or the NC, okay, which one has the inhibitor binding to the active site? If the inhibitor is bound to the active site, is that competitive or non-competitive? Competitive, beautiful, right? Because if you look, the inhibitor is bound to the active site and it's going to physically compete with the actual substrate. So that's competitive inhibition. Non-competitive is when you have the inhibitor bind to a different site on the enzyme. Here is your bonus question. What is the name of the other site that's not the active site that the inhibitor can bind? It does start with an A. Very nice, you guys. Great job. Allosteric. Good. So that's called allosteric regulation. Okay. So the active site is where the substrate binds. The allosteric site is another site kind of on the other end of the enzyme where the inhibitor will bind. Once it binds, it changes the shape of the active site. And now that substrate no longer fits. Okay. So competitive versus non-competitive inhibition. All right, Rashi, let's go ahead and see if they can do concept check 3. And then Jeffrey, if we could please go ahead and put it on the poll.
Which best describes what happens when an enzyme gets denatured? A, the peptide bonds between amino acids break. B, the enzyme unfolds as secondary, tertiary, and quaternary structures become disrupted. C, the primary structure unravels. Or D, only the quaternary structures separate. Looking at the poll, it seems like 85% of people are saying B, the enzyme unfolds, which is correct.
Good job, my loves. Great job. All right, so let's continue going through cellular energetics, okay? Everyone's favorite photosynthesis. We are just going to give you the shortest summary we can because photosynthesis and respiration could take 12 hours in itself. If you need a more thorough study guide, make sure you check out Fiveable's AP Bio section. It has a study guide for every single unit. So for those of you that need those more thorough explanations, make sure that you watch the videos. There's kind of reviews that you can do and question sets. If you're looking for certain slides and things like that, we have a little green button on the bottom of our screen that you guys could also click on. Okay. So photosynthesis basically is 2 different steps, okay? Step 1 is called the light-dependent reactions, and step 2 is called the Calvin cycle. It's also called the light-independent reactions. It's also called the dark reactions. So from the light-dependent reaction, essentially you have the special pigments that absorb wavelengths of light. Okay, so chlorophyll being one of the main ones. It's going to absorb different wavelengths of light within the thylakoids inside of the chloroplast. And it's going to go through a series of transformations. It's going to go through the electron transport chain. And we essentially are going to be moving the energy and using it to create 2 main products. Okay. The main point of the first step of photosynthesis is to create ATP and then another molecule called NADPH. Okay. So we're taking energy from the sun and storing it into a chemical energy, ATP and NADPH. And so what you need to kind of know essentially is how we get those 2 products. Okay. Now, a byproduct that happens is oxygen. Does anybody remember how do we get oxygen? What is split in the first part of photosynthesis that ends up producing oxygen? Water. Great job, you guys, right? So we split water to replace some lost electrons in the first step, and a byproduct is oxygen. So we are alive because of a random byproduct that plants happen to just diffuse out. Like, thank goodness, Lordy Lou, right? Like, I'm very grateful that they just have that byproduct. So that's not even the main point of photosynthesis. The main point is to take the energy from the sun and then be able to convert it into that chemical energy of ATP and NADPH. So here's kind of looking at one thylakoid, kind of like zooming in, and you can kind of see these two big like green things called photosystems. Photosystems are just these proteins that essentially are going to help house our pigments to allow us a way to organize absorbing all of that energy from the sun. Okay. And so when we have all these proteins kind of in the middle, just transporting electrons from one place to another, that's called the electron transport chain. And the electron transport chain is going to transport all those electrons. And in that process, we're going to create NADPH. Okay. So the electron transport chain is kind of what allows us to create NADPH. And every time we have an electron kind of going through this process, we're going to have a bunch of protons being dumped into this thylakoid space. Once we have a super high concentration of protons in the inside, it's then going to want to leave through ATP synthase, creating ATP. Does anybody remember, what do we call the movement of chemicals when they want to go from high to low? What is the movement of chemicals called? It starts with a C. What's the movement of chemicals called? There it is. Good job, Fifi. You got it. Chemiosmosis. Great job, you guys. Okay. So chemiosmosis is that electrochemical gradient of those hydrogen ions that have now, they've all been dumped in. And so it starts off with more protons on the outside of the thylakoid space. You basically shove it into the center. And then when it gets way too filled up, it wants to leave. Okay. So it's going to leave through ATP synthase, creating ATP. Beautiful. All right, so Rashi, let's go ahead and see if they can do another concept check. And then Jeffrey, if we can go ahead and put up the poll.
What process splits water, breaking a strong polar covalent bond to replace electrons? A, the Krebs cycle. B, glycolysis and the Krebs cycle. C, the citric acid cycle. Or D, light reactions. Wow, so it is overwhelmingly D, light reactions, and that is in fact correct. Good job.
Good job, you guys. We try to mess you up with those words. Great job. Okay, beautiful. We just went over that. That was the light reactions, okay? So what I want to briefly go over now is going to be like what happens once we have ATP and NADPH, okay? So what's going to happen in the next step in the dark reactions is it's going to be kind of on the outside of the thylakoid, still within the chloroplast itself. And we're going to take CO2 from the atmosphere, which comes from us, and we're going to fix it onto an enzyme called ribisco. And we're basically going to take the CO2, combine it with this bigger molecule, and kind of just mix it all up, okay? And in that process, we're going to create a little sugar molecule called G3P. Does anybody remember how many G3Ps are needed to make one glucose molecule? And yes, that's called carbon fixation. Great job. Ooh, that's kind of a trick question. It's 2, okay? 2 G3Ps are needed to create one glucose, okay? And yes, very good. The process I was talking about is called carbon fixation. This idea that we're going to fix carbon to our enzyme, ribisco, And then we're going to go through and we're going to rearrange the molecule to produce that little baby sugar, okay? And then we just have to go ahead and regenerate our original molecule so that it can then fix carbon again, and the whole thing happens again and again and again and again, okay? So in terms of big picture things, I want you to know that oxygen, ATP, and NADPH are produced in the light-dependent phase, and then CO2 is needed in order to form our baby sugar, okay? So that's the big things that I want you to know. All right. So next, we're going to go right into cellular respiration. And again, we're doing big picture things because we have a lot to cover tonight. And if you need more help on it, then please go into Fiveable AP Bio and you'll see slideshows, videos, study guides that will give you a lot more detail, or you can even watch some of the replays when we did it, okay? So photosynthesis and respiration are two processes that are going to be connected. You have one process that requires energy from the sun in order for it to occur, and it's going to store energy in the bonds. And then respiration is going to be releasing that energy into a usable form, okay? So what I want you to know is going to be, number one, who is able to produce each of the different steps? So we know that producers are able to do photosynthesis, but producers can also do cellular respiration, okay? We as a consumer can only do cellular respiration, okay? We cannot use the sun directly, even though that would be kind of cool, okay? If we could all photosynthesize, that'd be great. And so if you were to look at both processes, they're very similar. Essentially, you have one molecule transporting something to another molecule, transporting to another molecule, okay? So it's like a game of hot potato, right? So I'm going to take electrons from here, I'm going to transport them and move them here. So we're constantly going to be gaining electrons and losing electrons. So we have two scientific words for you guys to know. We have oxidation and we have reduction, okay? So oxidation, reduction. Between those two, oxidation, reduction, which one is going to be losing an electron? So when something has lost an electron, is it oxidized or reduced? Oxidation, very good, you guys, you got it. Okay, so oxidation is loss of electrons, reduction is gain of electrons. And there are kind of two silly ways of remembering it. I tell my students oil rig, oxidation is loss of electrons, reduction means gain of electrons. But I know that a lot of people also use Leo, okay? So whatever way you want to remember it, that's just going to be kind of like your trick. So don't get mixed up when you see those words. Yes, oxidation reduction is going to happen in photosynthesis and also in respiration. So it's something that will happen in both. Something else I want you to know is ATP versus ADP, okay? So ATP versus ADP. Which of those molecules are going to have 3 phosphates? Is it ATP or ADP? ATP, very good. It's adenosine triphosphate, okay? So the energy is stored in that last phosphate that is there. So if I were to release that last phosphate, it's going to release the energy. And now I go into ADP. If I want to store energy, ADP becomes ATP. And they kind of go back and forth. Okay, so we kind of say that they're coupled reactions. All right, cellular respiration. Okay, big picture things. You're going to get the most amount of ATP from aerobic respiration. Aerobic means in the presence of oxygen. So like right now, I'm going through aerobic respiration, okay? My body is maximizing the amount of ATP. If I were to start running, which I would not do, but if I were to start running, like some of you guys, I'm sure you guys run. If you were to start running, all of a sudden you're going to be starting like gasping for air. At that point, you're putting yourself through something called anaerobic respiration. You're depleting yourself of the oxygen needed for full maximum amount of ATP. So you're going to go through anaerobic respiration. So I want you to know aerobic respiration will give us a max amount of ATP, and that's going to be in the presence of oxygen. And then anaerobic means without oxygen. You'll still get ATP, but not enough for you to go ahead and like continue to thrive on that, okay? So you need to know the big picture steps of respiration. And the first step in both processes is called glycolysis. Glycolysis is an anaerobic process that is the first step in both of the different pathways that you could go. Glycolysis is going to produce 2 ATP. That's the big picture things. If you look at the word glycolysis, we have to break down glucose. You literally take a 6-carbon molecule and you're breaking him in half into 2 3-carbon molecules. So I want you to remember several rules of respiration. Number 1, every time you break a bond, then we're going to have some sort of electrons are released, okay? Every time you have electrons released, you have to have an electron carrier there to capture it, because that's the point of respiration, is to break all the energy out of these bonds. So our 2 electron carriers in respiration are NAD and FAD, okay? So NAD or FAD are there to capture anytime a bond is broken. So when we went from 6-carbon molecule to 2 3-carbon, we had to break a bond. NAD was there to capture it, okay? So he's going to capture that electron and then bring it inside the mitochondria. The next rule I want you to know is that every time you change the amount of carbons in a molecule, you end up changing the name of the molecule. And so welcome to organic chemistry. Okay, so it originally might have 6 carbons, now it has 3. Now it has a new name. Now we call it pyruvate molecules. Okay. The other rule I want you to know is that when you go down in carbons, you're going to be releasing a carbon as a CO2. And those are my rules of respiration. Okay, so anytime a bond is broken, NAD or FAD is there to capture those electrons. Then anytime you change the number of carbons, you have to give it a new name. And then essentially, anytime you go down or reduce in carbons, it's going to be released as CO2. Okay, so let's go through aerobic respiration. So glycolysis, a 6-carbon molecule gets broken into 2 3-carbon molecules. NAD is there to capture it. We now have 3-carbon molecules, so we call them pyruvates, and we get 2 ATP in that process. Then in the presence of oxygen, our 3-carbon molecules, there's 2 of them. They want to get pulled into the mitochondria, but they're too big. So we have to go from a 3-carbon molecule to a 2-carbon molecule. So we've released energy, there's NAD. We've gone from 3-carbon to 2-carbon, we've released the CO2. And now we have to add an enzyme in our new molecule called acetyl-CoA. This molecule is then needed for the next step, which is called the citric acid cycle. Okay. All you have to know about the citric acid cycle is that we're going to now take whatever energy was left in glucose, we're going to break it down and have nothing left. So all that energy and all that time we spent taking all the energy from the sun and storing it in glucose, it's all gone. There is nothing left, okay? We have now taken everything. Not a single part of that molecule is left, and it's been released as CO2, and all those electrons have been captured. We then are going to take all of those electrons to that very last step, which produces the most amount of ATP. And I do want you to know that, okay? So of all the steps, the last one produces the most amount of ATP. And so that one is called oxidative phosphorylation. It includes the electron transport chain. It includes chemiosmosis. It's basically the same process, just inside the mitochondria. So we get a lot of ATP in that very last step. Now, say oxygen is not present and we have to kind of stay outside, okay? So if we are going to have to stay outside of the mitochondria, we're going to go through 2 possible pathways depending on what organism we have, okay? We go through lactic acid fermentation. So essentially you go through glycolysis because that's the first step. And then we just kind of go through a series of enzymatic reactions that regenerate NAD just so we can continue going through glycolysis over and over and over. And in the process, we get lactic acid. Yeast and bacteria, they're going to go through alcoholic fermentation. So they still go through glycolysis. They still want to regenerate NAD. They still go through enzymatic reactions, but their products are alcohol and CO2. Okay. So I want you to remember that the point of anaerobic respiration is to regenerate NAD so that we can go ahead and go through glycolysis again and again until oxygen is present. All right, Rashi and Jeffrey, if we can go ahead and check them on their next concept check.
Which process of aerobic respiration produces the maximum amount of ATP? A, glycolysis, B, intermediate reactions, C, citric acid cycle, or D, oxidative phosphorylation? And this is including the ETC and chemiosmosis. And it looks like the majority of people, or over 95% of people, are saying D, oxidative phosphorylation, which is correct. Good job.
Good job, my loves. All right, we're going to keep on moving forward to Unit 4. So we have gone through Unit 1, 2, and 3, and Unit 4 is about cell communication, okay? So in terms of communication, cells can do long-term communication, that's called endocrine. It can be near each other, that's called paracrine. Self is autocrine, and then juxtacrine are going to be those times that those cells are right next to each other and have to communicate almost instantly. Those would be like your heart cells, right? So in your heart cells, you want to be able to have those electrical currents going through immediately. That would be juxtacrine type of communications. And so the two key terms that you need to know are receptors and ligands. A ligand is any molecule that is going to bind to a receptor. The receptor is like a little protein on a surface that binds to the ligand, and then it initiates some sort of communication. And this is how we can kind of communicate from the outside of the cell to the inside. And so what we do is we go through a process called a signal transduction pathway. And it's just a fancy way of saying receiving information from the outside and then causing something to happen, okay? So maybe we need to create more of a specific protein. So we're going to get some sort of signal, which is our ligand. It'll bind to the receptor. We're going to get some sort of conformational change in shape, and it's going to activate a whole series of enzymes that will then eventually make its way into the nucleus, and we're going to create something, okay? So that's called a signal transduction pathway. It's just how we can get communication from the outside to the inside. And so cells in general want to be able to have this feedback going all the time. And so there are two different types of feedbacks. There's positive feedback, and then there's negative feedback. Between the two terms, positive and negative, okay, which one means you have some sort of set point and you just move past it? You just keep going and you never come back. Is that negative or positive? Which one means a set point and you just keep going? Good job, Kiki. Positive. Good job, my loves. Okay. So positive feedback is when you essentially just have a set point and you keep going. So like fruit ripening, that's my example, right? You've never had a brown banana like go the other way, because if so, that would be kind of wild. And then negative is when you have a set point and you kind of go above and below, but then you always come back to that point. Very good. The cell cycle, okay? For the cell cycle, we know that cells are going to spend most of their time in interphase. That's just the cell growing. And then it's going to have to decide, is it going to die in cellular, that's called apoptosis, or is it going to divide? Remember, because we talked about cells can't get too big, and so it's going to divide, and that process is through mitosis. And we know that mitosis will be prophase, metaphase, anaphase, telophase, and then that very last step is cytokinesis. So that's kind of the basics of the cell cycle, not too complicated. In terms of the terms that students tend to get confused with is the difference between a chromatin and a chromatid, and then essentially a chromosome. So I want to kind of go over those words with you. So if your cell is just growing in interphase, like legit just growing, then it looks like a bowl of pasta, and that's called chromatin. It's just like there. It's like blah. Okay. But right when we get ready to divide, we want to like organize it so that when we're moving it, we don't like mess anything up. And so you kind of like coil it up around these proteins, and now it's coiled, and that's called chromatid. So chromatin is uncoiled DNA, and then chromatid is when we're going to coil it up. When we're getting ready to divide it, we want to make a copy of that, right? And so now those are called sister chromatids or chromosomes. So like if I were to ask you to draw a chromosome, you would draw the letter X. That's what a chromosome is. And each individual arm is a chromatid, and then uncoiled is chromatin. Okay, so here's just a fun little break question for you. What would you fear the most? And I think we have a poll on this too. Would you fear the most, A, a pit of snakes and spiders, B, not being able to say anything but the truth, always, nothing but the truth, or C, public speaking? What would be the worst for you? Looks like a tie between A and B. All right, fair enough. Okay, a couple of you chose C. Okay, good to know. All right, we now have a whole review of units 1 to 4. So now I'm going to kind of hand you guys over to Rashi, who's going to do a couple tips and tricks, and then we're going to do some practice FRQs.
Okay, so first let's go over the tips. I'm not sure when the tip section vanished on my screen, but it's the list on the left. The first one is make sure you're timing yourself throughout the exam and bring a non-smartwatch. Remember, things like Apple Watches or the Samsung Watches are not allowed. And keep an eye on the clock, because the worst thing you can do is maybe if you get stuck on a question, a really hard question, you're just sitting there and you're stumped, and then you look up and it's been like 10 minutes on this one question. Don't bother wasting your time like that. Instead, move on past the question and come to it towards the end. The second one is know your task words, and we'll go through those on the next slide more properly. The third one is don't include fluff. You don't need to restate the prompt for your FRQs. Make sure you write in complete sentences, but there's no point in including information that they've already given you in the prompt itself. And that kind of ties into the fourth part, which is eliminating extraneous information. If it doesn't ask you to define something, don't define it. Just list the word and move on. You want to make sure you cover all parts of the question, but also save yourself time by only covering what's needed. And some tricks. Answer the short, easy questions first, especially on FRQs where they ask you to identify or propose. And a lot of you guys have asked this. You can answer the FRQs out of order as long as you're clearly labeling which part and which FRQ you're working on. So if you get stumped on one part, move on, skip ahead to the next part, and come back to it at the end. Use complete sentences, otherwise your AP graders will not be happy. And to label all your responses clearly. I think the worst thing would be like knowing you have the right answer, but you put it as maybe question 2 instead of question 3, and now it's not being graded at all. So make sure you guys are using complete sentences and labeling everything clearly with the question number and the question part as well. And then these are the task verbs that we mentioned. So you'll see these throughout the FRQs, and these are very common. The first one is describe. You want to provide information on what is happening. And it's similar to explain, but explain also involves describing how something can be happening. Then there's identify, which is, of course, a one-word explanation. Justifying is to prove something, and you'll see this a lot in justify this claim. Researchers have made this claim, justify it. You have analyze. Look at the situation and describe what's happening. But here it's asking you to provide your own insight into this or your own interpretation. For example, analyzing a graph and interpreting what the graph is describing. And finally, you have predict. Make a prediction about what you think will happen. And if it says predict, include as much evidence as you can to back up your claim. Graders know exactly what they're looking for. So if you don't match it, they can't give you credit, which means that you want to make sure you guys are following these task verbs very closely. And some more FRQ/question strategies. The first one is use the KISS method, and the second one is remember ATP. So imagine the AP test is like an Easter egg hunt, okay? The graphics have all the answers, but you don't want to get tripped up. Like think of the answer choices as like your friends telling you, oh, the Easter egg is there, the Easter egg is here. You want to think about first, where do I think the Easter egg is? Then listen to your friends. So in this case, answer the question before you look at your choices. Then there's the KISS method, which is keep it simple, silly. Wash away any big words. The AP exam is going to provide you with a bunch of definitions, a bunch of like weird enzymes or maybe proteins that you've never heard of, but ignore these. Ignore the big words and look at specifically what it's asking you to answer, because that's what's most important. And then you have the ATP method, which is answer the prompt. Do not go overboard and start defining things to the point where you might end up accidentally contradicting yourself. You just need to answer the prompt. And the other side is make sure you're providing enough information that every part of the prompt is being answered completely. Remember that these FRQs are not recall, and that's what makes it so much more difficult. They're asking you to apply the knowledge. But we have full faith that you guys know the knowledge. So remember to stay calm so that you can actually properly apply it. Don't just list out everything you know. And now we're going to go over into some multiple choice practice. So this question says, a common laboratory investigation involves putting a solution of starch and glucose into a dialysis bag and suspending the bag in a beaker of water as shown in the figure below. And the left figure, as you can see, the bag has starch and glucose and there's water outside. And then in the right one, the figure has changed. The investigation is aimed at understanding how molecular size affects movement through a membrane. Which of the following best represents the amount of starch, water, and glucose in the dialysis bag over the course of the investigation? And we'll give you guys a minute to answer this. And so the answer is A. If you look closely in the graph in A, you can see that the water increases over time, the starch level remains the same, and the glucose decreases. This is because in the original dialysis bag, there isn't any water. So the water is going to, because the water is going from a high concentration to a low concentration, is going to go through the dialysis bag into that bag and increase the concentration of water in there. However, if you look at the original diagram, the glucose is only inside of the dialysis bag. And so it'll have the opposite reaction and go from the dialysis bag into the water until the concentration gradient is about the same. Starch cannot move through a membrane. So the level of starch is going to stay constant inside of this dialysis bag and won't be impacted by the moving of the water and the glucose. It is estimated that oxygen production first evolved in photosynthetic prokaryotes approximately 2.7 billion years ago. The first photosynthetic prokaryotes are presumed to be similar to today's cyanobacteria. Which of the following best supports the claim that photosynthetic prokaryotes were responsible for the oxygen in Earth's atmosphere? A, the light reactions of photosynthesis split carbon dioxide into carbon and oxygen. B, the light reactions of photosynthesis split water into hydrogen ions and oxygen. C, the Calvin cycle splits glucose into carbon, hydrogen, and oxygen. Or D, the Calvin cycle splits water into hydrogen ions and oxygen. So we'll give you guys another minute to answer this. I'm seeing a lot of B's in the chat, and that is correct. The light reactions of photosynthesis split water into hydrogen ions and oxygen. And this one was just kind of recalling that information from what a light reaction does. Notice how, even though it talks about a lot of like cyanobacteria and evolution within the prompt itself, or within the question, the background information itself, the prompt is really just asking you which one of these 4 facts is correct.
Great job, Rashi. Thank you so much for your help with the multiple choice questions. All right. I know that we're going through. I know you guys are tired. We're going to go ahead and we're going to take a 3-minute break. Okay. I want you guys to have a nice stretch. I want you to grab a snack. I want you to do whatever you need for 3 minutes. And then we're going to come back and we're going to go right into Hardy-Weinberg stuff. Okay. We're going to kind of review Hardy-Weinberg. So you got 3 minutes. All right, my loves. Here we go. Hardy-Weinberg time. Okay, so Hardy-Weinberg essentially is an equation that we use to predict changes in allele frequencies within a population. Okay, and so if we wanted to go to a population and look at all the alleles that are there, then we want to be able to predict the frequency of those alleles. And so if we went to this particular pond and we looked at the frogs and we calculated their frequencies, and then we came back, let's say a year from now, and a year from now, and a year from now, we want to be able to track those changes in those allele frequencies. And Hardy-Weinberg basically came up with this equilibrium. They said that the frequency should remain the same. And if it does, then we say the population's at equilibrium. So if a population is at equilibrium, we have 5 assumptions that are being held: that there are no mutations, random mating, no natural selection, large population size, and no gene flow. But if we look at those allele frequencies and they are changing, then that means evolution is happening. So in order to solve these problems, I try to simplify it like as much as possible. You essentially need to know a couple things, okay? The main equation that you need to know is P plus Q equals 1. Like literally that is it right there, okay? P plus Q equals 1. It's going to be in your formula sheet, so you don't have to memorize it essentially. But in my head, I always remember P plus Q equals 1. Then you have to know what that stands for. P stands for the frequency of the dominant allele. So the big letter, just the big letter. So in this situation, I'm just calling it big A. Q stands for the frequency of the recessive allele, little a. And so your job is to look at what are they giving me, and immediately you should be solving for P and Q. That is like your go-to in terms of steps. So again, what are they giving me, and how do I solve for P or Q? Because P and Q always equals 1. Once you know P and Q, then essentially you just kind of like plug and chug, okay? So I always make these little variable tables because I know that I need to essentially solve for 5 things for every problem. This is just what I do. This is my method. People that are really good at math and patterns, you probably don't need to do this. But I like to go ahead and create these tables. So I make a little table and I put P and Q. I make another table for that says P squared, 2PQ, and Q squared. P squared stands for homozygous dominant. So that would be like big letter, big letter, big A, big A. 2PQ represents the genotype of the population that's heterozygous, big A, little a. And then Q squared represents the population that is homozygous recessive, little a, little a. And so you're always going to be given one of these variables, and then your job is to figure out the rest, okay? Now, the hardest part that students have is always determining, are they giving me the Q or are they giving me the Q squared? Couple things to look out for. If it says alleles, then they're giving you a P or Q because P and Q are your alleles. But they will always say the frequency of the dominant allele or the frequency of the recessive allele. If it's talking about genotype, then it's giving you one of these. Okay, so let's kind of do a problem together. So the tiger moth has the following genotypes. Oh, I'm sorry, did the closed captions come off again? I'll put it back on. There we go. Okay, so here are the tiger moths. It has the following genotypes, and we have big A is 500, big A, little a is 70, little a is 30. Okay, so I want you guys to try these problems. The only way that you're going to get good at them is figuring out this pattern. Now, in these boxes, you're welcome, Aubrey. In these boxes, I want you to know that you are to always put in a frequency, which means a decimal. It's never going to be a whole number, okay? So they've given you these numbers. So we're going to go ahead and turn them into frequencies. So P squared equals 540 over the total, which is 640. 2PQ is 70 over 640, and Q squared is 30 over 640. Again, I just had to turn it into a frequency. I couldn't just put in a whole number, okay? So once you put it in the calculator, we end up with these numbers. Now, I told you that your job is to solve for P and Q because P plus Q equals 1. That's the easiest one to remember. And so what I recommend doing is I always use Q squared. So I just figured out Q squared equals 0.05. Then you want to just isolate Q. So take the square root of both, and that's going to give you a Q value. Then you're going to take that number, and then you're going to subtract it from 1, and that's going to give you your P. So in the chat, tell me what you think P and Q are. Given Q squared equals 0.05, take the square root of Q, so the square root of 0.05, that gives you Q, and then do 1 minus that number, that gives you P. All right, very good, you guys. So P is 0.78 and Q equals 0.22. Great job. Boom, done. All right, let's try another problem. So again, I would draw my variable tables again, or if you're really good at math, you're probably like, I don't need to do this, assuming I got this. In a population of beetles, 60% have the homozygous recessive allele for color. So then I go, what did they give me? They said 60% are homozygous recessive. So they're giving me Q squared. So Q squared equals 0.60. Okay, now go right into solving P and Q. So Q squared equals 0.60. Isolate Q by taking the square root of Q. That gives you Q. Then you're going to do 1 minus Q. That gives you P. Yes, you also could have done P squared and then taken the square root of P to get P. Absolutely, you could. I just always go to Q squared just by default. Okay, once you have P and Q, then you just plug and chug. So once you have P, then you just do P squared. And then to get 2PQ, you do 2 times P times Q. Then you have your entire chart filled. But this is not where you're done, because you have to answer the question. The question says, what percent of the population is heterozygous? Heterozygous is going to be 2PQ. So we got 0.35. But the question says the percent. So you have to answer it as 35%. Okay, so 35%. All right, let's try another one. This one's going to be a little trickier. In a population of 500 total moths, the frequency of the recessive allele is 0.3. Beautiful. Look at that, you guys. They gave us Q. Okay, now take a minute and you guys figure out the rest. So they gave us Q. I got you, Sophia. So they gave you Q. Figure out P. Plug and chug. And let me give you a little time to see if you can come up with the final answer. Okay, so we have 0.3. We know that P plus Q equals 1. So we would say 1 minus 0.3 would give us the number that we need, right? And then we're going to go ahead and square P, do 2PQ. So now we end up with something like this, okay? But now we have to figure out the final answer. So it says, how many individuals are homozygous dominant? Homozygous dominant is big A, therefore is P squared. Okay, so it's going to be 0.49. That's homozygous dominant. But they want to know how many individuals. So they're not saying the frequency is 0.49. They're not saying what is the percentage, because you would say 49%. They're saying how many individuals. Okay, so if my total population is 500, figure out 49% of 500. And then that should give you the total number. So I'll give you a few seconds to kind of figure out how could I do 49% of 500, and that would be our final number. $245. Good job, you guys. Great job. I know that was easy for some of you, but some of you guys are better at math than others. So it's important that we're all supportive of each other and help each other out, okay? And so this is kind of like a tricky question. But if you could just, every time I get a Hardy-Weinberg, I always say to myself, what did they give me? And then I'm going to plug it in. How do I solve for P and Q? And then I plug and chug, and then I say, what is the actual question itself? Okay. And so that's kind of just like the basic formatting of how you're going to solve Hardy-Weinberg. Okay. So draw your variables table always. They will give you the, in the formula sheet, the equation for Hardy-Weinberg, but I just draw my variables tables because that just always helps me out. And then I'm going to figure out what did they give me. Then I'm going to calculate all the variables that I have. And then I'm going to ask, what are they asking me? Okay. All right. If you want more problems, if you guys go to the Crayons AP Bio site, it's going to have a whole bunch of practice problems for you guys to be able to do more of that if you would like to. All right. So I have a question for you. What would you rather have an infinite amount of? The ability to heal, money, or intelligence? What do you think? What would you want to have an infinite amount of? All right, I kind of feel like we're all over. That's interesting. A lot of people say intelligence. I think I would choose the ability to heal. All right, you guys, thank you so much for being willing to share. And I have to introduce you to somebody. So I'm going to stop sharing my screen for a second. Okay, I am going to introduce you to Miss Kari. Miss Kari, do you want to come join the screen with me? Look, there she is. Hi, Kari.
Hi. Hi, everybody.
All right, so at
Nice
this
to hear.
point, I'm going to hand you over to our amazing co-host, Ms. Kari.
Okay.
And she's going to go over the rest of the units with you, so she'll help answer. They had some questions about the Tata pox and things like that, and of course, you're going to do it. And then, Kari, they are begging for Samasoa Sama.
Oh, yeah, of course. Some practice problems. Or should I just have the music play? We don't even have to do the practice problems.
Yeah.
I think that's
all right, you guys. So Miss Kari is going to take it over. I'm wishing you guys the best of luck. I 100% have faith in you. I know you can do this. And Jeffrey and Rosh are going to be with you the whole night as well. You got this, okay? And you will end your night with some samaso. See, look, do you see them, Kari? They're all about it.
Oh, I know. It was like that the last 2 nights, too. It's hilarious. It's so
all right,
funny.
you guys. I am signing out. Kari, they are yours. Good luck, you guys.
Bye. Good job. Thank you. Yay! Oh, you are all so funny. Well, hi everybody. You have reached the halfway point of your 3-hour cram for tonight. If you have not been here on the previous nights, my name is Kari Parnan, and I love AP Biology. I love teaching, and I'm excited to be here with you. And it was fun. I mean, I feel like I've gotten to know a few of you already just through the chat over the last few nights. I'm going to start with actually a 3-minute break for all of you. I'm not sure if you've had a break recently, but that's what we're going to do. And so during your 3-minute break, I will put on the music if I can find it. So take 3 minutes, go to the bathroom, get a snack. I know it's 8:30 my time here in Michigan. So I'm going to start the timer and hopefully you can all see it. So let me get that going for you. And I'll see you in 3 minutes. There we go. And now we'll say 3 minutes. Sorry about that. All right, I'm gonna come back in just a second. I cannot believe you couldn't hear the music. I'm so sorry. What happened? Let me get moving here. Okay. I'm so surprised no one could hear it. Hold on, let me get myself back over to the side. All right, we will get that music going in just a little bit. I know, it's so good. 3 more minutes. I see some comments in the chat about going over graphs, and we do have some time for that. So just give me one moment. And actually, I just realized, Rashi, you're the one that's introducing this unit. So go ahead, hop on.
So for the past 1.5 hours, we've covered units 1 through 4, and now it's time to tackle units 5 through 8. So we're going to go over the top 10 big sections that we feel have the most information and are the most relevant for the AP Bio exam. And on the next slide, you guys will see kind of like the list of from the AP Bio CED. And if you guys can see the boxes, they're a little hard to see. That is all the information and the percentages of each unit that we'll be covering. So we're covering over like 50%, I think what for unit 6 is everything except for like 1, 2 small items that we're covering. So good luck.
Thanks, Rashi. Okay, team, let's do this. What I tried to do was I'll cover some stuff that we did the last few nights, but I wanted to throw in some things that we also did not get to, and then have some free time potentially to go over some of the most asked for items right now, which I already see is graphing and water potential. So I want to ask if you remember mitosis and meiosis and what their similarities are, or what their, sorry, let's start with differences. So I'm going to put up mitosis, and I want you to think about what's different about meiosis. If you were to list all the differences there, what would you write? And we'll do the similarities after that. So sorry about that it's showing similarities right now. So think about, I gave you mitosis, and now you just kind of have to think about, well, what's different about meiosis? Good. I'm seeing some good words. I'm seeing 2 divisions, creates gametes, haploid. All right. So many of you are getting it. Excellent. So yes, meiosis is creating gametes or sex cells, so the sperm and egg. They are creating haploid cells, and then they are going to be genetically unique. And the way we get to those genetically unique haploid cells is by 2 sets of divisions. So you're going to have prophase, metaphase, anaphase, telophase, and then you do it all over again. Prophase 1, metaphase 1, prophase 1 is the first set, and then prophase 2, metaphase, anaphase, telophase 2. Now I want you to think about what the similarities are, okay? Because a lot of the times it's easy to go, what's so different about them? But what do mitosis and meiosis have in common? Okay, I see some people saying they both produce daughter cells, the actual stages, right? Prophase, metaphase, anaphase, telophase. Good. Excellent, excellent. And the other thing I want you to think about, and here's a picture, right, of those phases. I've got prophase, metaphase, anaphase, telophase. But I also want you to think about what the chromosomes are doing. So that's why you don't technically need to have any of those phases memorized. You need to be talking about what are the chromosomes doing, right? So in prophase, they're already going to be condensed into chromosomes, right? And then within those different phases, what are the chromosomes doing? Are they moving toward the center? Are they moving toward the pole? And really the movement of the chromosomes is the most important part, because without the movement of those chromosomes, we would end up with really disorganized daughter cells that either don't have the correct number of chromosomes or maybe have too many or too few. Some other things they both do is spindle fibers, and then they both experience sister chromatids separating. So if I have a quote here in purple, it says mitosis and meiosis are similar in the way that chromosomes segregate. Take a second. Would you be able to explain what that means? They're similar in how chromosomes segregate. Go ahead and try to think about how you would explain that. They're similar in how chromosomes separate. Okay, I see someone saying the way chromosomes are divided or pulled to opposite sides. They both do anaphase. Good. First, they have to line up on the metaphase plate. Good. Good. Okay. Excellent. So yes, they're similar in how they segregate. They get their chromosomes lined up, and then they actually separate the chromosomes after that, which some of you named there as anaphase. So really good job. Now I want you to look at a diagram, and I want you to tell me what is happening here. You can either tell me what phase it's in, or you can just be just a really simple description. What's happening here? Focus on the chromosomes. Don't just tell me the name of the phase, right? Because just naming the phase from a picture, that's not college level question or answer. Yes, I see some people saying it. So yes, it's metaphase, but specifically, it's your sister chromatids lining up on the metaphase plate. When I was learning this as a student, I remember feeling really confused about sister chromatids and homologous chromosomes. And so if that's something that's still confusing you, try to picture this image of sister chromatids lining up here in the metaphase plate. So this would be metaphase and mitosis. What about here? What's happening here? Take a look at this picture. What is happening here? Ooh. Okay. I see some of you are saying crossing over, but be careful. Crossing over has already happened at this point. Crossing over has happened here. Nice. I see some people saying homologous chromosomes. Excellent. So this is a little bit different from the last slide because one, we see, one moment. Sorry, technical difficulties. I'm trying to get my laser pointer. Okay, so this looks different than the other one because we've got two chromosomes here right next to each other. And notice how these are the same size and these are the same size. So those are likely homologous, right? So this might be chromosome 1 from mom, chromosome 1 from dad. And then this might be, let's say, chromosome 3 from mom, chromosome 3 from dad. And some of you said, hey, this is crossing over. However, the crossing over has already happened. And I apologize if the red and blue are not showing up on your screen, but what we're seeing here is that this red chromosome, for example, has some blue fragments from this blue homologous pair. So chromosome crossing over has already happened. But a lot of you were correct. You said homologous chromosomes are lining up at the metaphase plate. And then I want you to consider this question. So let me throw together a quick poll. Read the question while I make the poll. And it should show up at the top of your poll. So go ahead. The question says, is this meiosis or mitosis? And how do you know? A says meiosis because there's no evidence of crossing over. B, meiosis because there is evidence of crossing over. And C, mitosis because homologous chromosomes are preparing to separate. All right. You are all amazing. Look at that, 99%, absolutely correct. Your answer is going to be B. And this isn't exactly, the length of a test question that you'd see tomorrow, but I did want to point something out. Notice how A and B are saying something very similar, but the difference is this word, right? So this first part of A is right. And then you see the word crossing over, but it says there's no evidence of crossing over. And there will be a lot of that tomorrow where two answers maybe sound really similar, but it's going to be like a word or two that actually contradicts what the first part of the question says. So be really careful of that. If you were to all of a sudden look at this and be like, meiosis, I know it, but you have to read the whole answer. okay, because they're sneaky like that. They want to make sure that you're reading the entire answer, because that's the difference between a 5 and a 2, right? Is that someone really read their answers and really thought about, is this entire answer option correct, or is only part of it? All right, so that's just a little test taking strategy that is always really helpful. Moving on to meiosis and genetic diversity. I want you first to think about what is genetic diversity, because it's a word that you hear all year long and might even be something you heard in middle school, but what does that even mean? So think about that. Ooh, I like some of the answers I'm seeing. Diversity in alleles, diversity in genes, variation of the alleles, differences in the genetic code. All those are fantastic answers. This is just my simple answer, right? The variety of the alleles in the population. But what a lot of you said was wonderful and accurate as well. So this picture is just kind of showing it as zoomed in chromosome here, where this one chromosome is looking blue or purple to represent one variation of the allele. And then the other one here is kind of like a tan colored. And that's representing that we've got 2 different alleles for potentially the same gene right there. And so the variety is key. This is a study that's actually from the real life. In the before the 1900s, the Florida panther covered this entire blue area. They were doing really, really well. However, their habitat was restricted in the 1980s to this tiny little dot over here. And because of that, instead of having a wide variety of alleles, all of a sudden there was not much genetic diversity. And so I apologize again if you can't see the variation in color here, but this first original population, the variation is represented by 4 or 5 different colors. And so those different colors are a model of, there's a lot of different alleles, right? And then this new population that was fragmented in size had really low genetic diversity, which is represented by just only 2 colors of the panthers here. Okay, so not actual color of panther, it's a model. But what ends up happening is almost like an inbreeding situation. And if you've ever covered any kind of inbreeding situation where 2 really related individuals have offspring, that tends to lead to health issues or increased chance of mutation and chromosomal abnormalities. And so this population just kept getting smaller and smaller because with their low genetic diversity, they really struggled with disease. They struggled with high mutation rate. And what ended up happening was that they had the same species of panthers that lived in Texas, and they actually brought them over to Florida to mate with the Florida panthers. And so now the genetic diversity increased greatly. And so it actually really saved that population, because eventually that could cause a complete decline and eventual endangered species extinction situation. So big story, you want a lot of genetic diversity. It's a good thing. Next thing I want you to think about, and you can either think about it, jot it down, or say in the chat, explain how the process of meiosis generates genetic diversity. Hopefully you thought about crossing over or recombination, an independent assortment, and random fertilization. And then hopefully your mind was starting to also think about when those things happen. So crossing over and independent assortment both happen in prophase 1, or they start to. Okay, and then I think Rashi's going to come in here and do a couple practice problems with you all.
During anaphase 2 of meiosis, a spindle fiber breaks. Describe the outcome. A, one daughter cell with an extra chromosome and one daughter cell missing a chromosome. B, two normal daughter cells, two daughter cells with missing chromosomes. C, two normal daughter cells and two daughter cells with extra chromosomes. Or D, four daughter cells with an extra chromosome. And if you guys click on the polls, there should be a poll open right now for you guys to go and vote. And it seems like the majority of people are saying A, one daughter cell with an extra chromosome and one daughter cell missing a chromosome. And that is in fact correct. Good job.
All right, thank you. Also, I want to let everybody know that I edited some of these slides like an hour and a half ago because we had some changes happen. And so there is a typo because I was going too fast, like humans do sometimes. This is supposed to say metaphase. Sorry about that. So independent assortment happens during metaphase. It's when your homologous pairs line up. And if you need more help with that, go ahead and ask in the Q&A and we can try to make some time at the end. So sorry about that. Okay, one other thing I want to talk about before really moving into genetics is fertilization, because I think it's one of those terms that just gets thrown around and assume that everyone can explain it or describe it, but maybe failing to use the words that we're using in this class. So try to describe the process of fertilization, mentioning the following words: gametes, haploid, and diploid. So during fertilization, haploid gametes combine to form a diploid cell with the correct copies of chromosomes. So it's a pretty simple explanation, but just sometimes throwing in those vocabulary words in something that makes sense can be a little bit difficult. So I tried to keep it concise to also model how you should answer your FRQs tomorrow. So especially if they give you that bolded word, describe, keep it really simple, right? And make sure you're answering the prompt. Remember 2 nights ago, ATP, answer the prompt. So I made sure that I mentioned the process. I use the vocabulary words mentioned in the correct way. We're going to step into Mendelian genetics and just start with a practice problem. I've got 2 traits that I'm looking at. So this is a dihybrid cross, and it says freckles are dominant to non-freckles. Dimples are dominant to no dimples. Determine the phenotypic and genotypic frequencies for the following cross. So take a moment, take a minute, maybe 2, to work through this. Okay, so I'm going to try something that I haven't tried yet on my computer. I'm going to see if I can write on my screen because sometimes that is most helpful. So the other night, some students talked about how their teachers taught them either the FOIL method or the divide and conquer method for doing a cross like this. So yes, I'm going to absolutely show how to do it. I'm just going to get my annotations going. And if it pulls up in a different screen, I'll just share a new screen. Hold on one second. Hold on. Sorry, I'm going to figure this out, I promise. Because I was really excited that I was able to do this for you all. Here. We're going to do 5 practice problems while I figure this out. How about that? And then you can listen to your song. See if it works. Put the link in the chat. There you go. We'll do a couple questions while I try to get my text to work. You can answer the questions too. Excellent. Insoluble in water is lipids. Next question. Good job, ribosomes. Yes, they are conserved. Very good. Good job. Enzymes are proteins, so they're made up of amino acids. Excellent. So this is the commonly mistaken one. So in meiosis 1, first the homologous pairs have to separate. And then in meiosis 2, you get your sister chromatids to separate. So homologous pairs first, and then sister chromatids second. Last question. Excellent, ribose. Great job, everybody. All right, I'm going to get rid of that. And we're going to pop back over to my screen. Whoopsie, I messed it up. But I have good news. I figured out my annotation thing, so I will be able to walk you through how to do this problem. So let's just wait a second to make sure that my screen pops up here for you. Loading. There we go. All right, back to Mendelian genetics. And I saw some conflicting answers in the chat, which is why I want to take the time to do this, because it's an easier thing to get when you're confused. And I bet there will be a problem like this on there tomorrow. So it's an easy reach for all of us to go for. Okay. Your teacher might have taught you like a FOIL method, or really, if you know meiosis well, that helps you here, because we look at how, for example, with this one trait, right, for dimples, this parent can't give each of those alleles to their egg or sperm. They have to separate, right? That's what law of segregation is from Mendelian genetics. Like, you can't, unless there's non-disjunction and it happens, but we create the Punnett square knowing that those alleles or our letters have to separate. And so it's really just a matter of, if you think of foiling from math, you look at all your options. So I would look at, for example, how my dominant D could pair up with my dominant F, and it can also pair up with my recessive F. So just like how I said the dimple alleles have to separate, the freckle alleles have to separate. And we can put those two together because we're assuming that that nothing is linked. Okay, so I'll write out my options here. So I have big D, big F, so dimples and freckles. And then we have the dimple allele, the no freckle allele. And then we look at our no dimple allele and how it could potentially be paired up with the freckles. And then it could also be paired up with the no freckles. So that would be from parent one. Those are going to be your allele options that could be given to the next generation. And then this other parent, they only have one of each allele. So you don't really need to FOIL because there's only one option. There's only going to be your recessive allele. Oh, that's a really bad deal. Sorry, is that an eraser? Nope. Yes, it is. Wonderful. So no dimples and no freckles are your other options there. And I apologize that this is going to be not the greatest Punnett square box. So I'm going to type the rest. All right. So then you put your alleles together and I say, okay, well, parent one and parent two, dominant, recessive. So those alleles drop down. And so that's one genotype possibility. The next one, and again, some of you are like, I know, just move on already. But again, let's be patient and let's be kind that this is something that can be helpful to a lot of people. And even if it's not helpful to you, that's okay. And you can just bear with us because that happens in life sometimes. You'll be in like a meeting for work or something like that. And you're like, oh my gosh, I learned about this a year ago, but some other people haven't. All right. I've got my genotypic layout here. I wouldn't even call these frequencies yet because I haven't put a number to them, right? So if the test is asking about genotypic frequencies, you'd have to list each of these separately and say, you know, I saw some people write 1 to 1 to 1 to 1, which is correct. Or you could write it as, you know, 25% chance of you know, this genotype, 25% of this, and so on and so forth. But now you want to think about what are the phenotypic frequencies, and you'd have to list those out specifically. Okay. So again, I'm going to use the text box just so that you don't have to look at my handwriting here. But what I would say is, you know, 25% would be dimples and freckles. So having both dominant traits. 25% would have dimples, no freckles. 25% would have no dimples with freckles. And 25% would be no dimples. Oops. No dimples, no freckles. Okay. How are you feeling? I'm going to make a quick poll. Did that help? Oopsie. So if you could answer this poll, just so I could get some immediate feedback if going through something that detailed helped. Oh, yay. I'm so happy. Can one of the student hosts help me make my screen look big or push me off to the side? Why is that happening? Well, thank you. I've 5 nights and I haven't figured this out. I'm so embarrassed. All right. Remember, when you get a problem like this, take a deep breath. It's just a puzzle, right? Look at the alleles, look at all the options, put it in a Punnett square, and read the prompt, right? Read it carefully, and you're all going to be just fine. So excellent job. I'm really happy. Wonderful. All right, let's see here now. Oops, nope. All right. I'm going to skip the sex-linked practice. I'm going to do this one instead. So another Punnett square you might be expected to do is looking at sex-linked traits. Now, generally that's going to be X-linked. It's going to be on the X chromosome. If they do put on the Y chromosome, roll with it and put the trait on the Y chromosome, right? In this one, it says one eye color gene in fruit flies is X-linked with a recessive allele giving a white eye and a dominant allele giving a red eye. If we have a white-eyed female bred to a red-eye female, describe the expected offspring. I want you to first think about what are the genotypes of these parents. So take 30 seconds and figure out what are the genotypes of the parents, because we can't do a cross until we have that. We are going to do chi-square at the end. Yeah, you bet. So while you're thinking about the genotypes, somebody asked, how can you tell if it's sex-linked from a pedigree? Generally, it's going to affect males more than females. Alrighty, hopefully your genotypes look something like this. So XY for dad, he has red eyes. And so it gave you that right there, that it's capital R. Mom has white eyes. So she's XX with both recessive alleles attached. All right, so you want to set up your Punnett square. Go ahead and do your cross, and then figure out what the offspring ratios are. I see somebody's asking about the 2013 AP Bio exam. That is before the most recent rewrite. So I think it was 2016 or 17 where they changed some things around as well. And even the format of the exam is a little different. And there's a lot less nitpicky things that you need to know, which is really nice. It's really problem solving. Tomorrow is going to be about reading the problem and working through it using critical thinking and not giving up. Okay, I see some people already giving some offspring ratios, so awesome. So hopefully your Punnett square looks something like this. And there's always several ways to describe the offspring if they're not going to give you something really specific. So you could talk about it separately that, 100% of the males will have white eyes and 100% of the females will have red, because even though they're carriers, remember it's a dominant trait. So there we are. Or you could talk about genotypic frequencies, right? So sometimes they ask for phenotypic frequencies and sometimes they ask for genotypic. The reason why it goes from 100 to 100 to 50 to 50 is that in that first phenotypic frequency part, I was separating X, Y, and X's, so males, females, and that's why. Whereas just genotypically, I just kept it a little bit different, okay? Alrighty. I've got a gene mapping problem, and we don't have a ton of time to let you really work through it, but I wanted to expose you to it just in case. The female parent is not heterozygous because she had white eyes, and it's a recessive trait. So she has to have both. Anyway, so I wanted you to at least be exposed to gene mapping, and you might get something like this because it's about gene linkage, and it is mentioned in the CED. So this is talking about how during crossing over, if genes are located on the same chromosome, the likelihood of crossover can be influenced by how close they are, okay? So for example, like if my finger is a chromosome and I have a gene way up here and a gene way down there, the chances of them crossing over and going to a different chromosome is high because they're far away from each other. But if you have genes that are super teeny tiny close to each other, the chance of them doing that would be really low. So they might give you recombination frequencies, which basically means like how often were those two things swapped to a new chromosomes? How often were they there? So I always draw out a line to start to represent my chromosome. So it's telling you there are these 4 traits, A, B, C, D, and they're all on the same chromosome. All right, so I start by making just a line across. And then I go to, okay, who has the most crossover? So that percentage is about crossover. Looking at the data, A to B has a high crossover. So I'm going to put them further apart from each other because they're likely to cross over if they're further apart. So I'd put A and B further apart. And then I'd say, okay, well, D to A is 33. So maybe D is a little bit closer to A, right? By 2%. So like 2 little notches. And then continue kind of problem solving. What's next? Okay, well, A to C is 25%. So maybe C is just a little bit closer, right? So we have B really far away from A. We have D a little closer. We have C a little closer, and so on and so forth. So like I said here, right, we have A to C, which is about 20, where is it? Sorry, A to B is 35 away. And then we have A to C, 25 away, right there. You really start to piece it together when you realize that D and B are 2% away. And then B to C are 10%. And I've never, as a student, been able to solve this on my first try, right? It's almost like you try one way and you see if the percentages work out, and then you try it another way to double check. It's another just puzzle. But I want you to know that if this is on the exam, it's going to be one thing, right? It might be like a tiny part of an FRQ, or it might be one multiple choice question, okay? So if there's anything, and I said this the other nights too, if there's something like this where you're like, I never learned this, this is the first time I've ever seen this, don't worry, it's okay. Like, do not freak out. It's going to be okay, right? But I just wanted to at least expose you to it tonight, all right? I know I'm taking forever on this unit, but we're going to keep going, okay? I'm moving through with DNA structure. I just want to talk about, what the nucleotide is, how it's the monomer of DNA. It's composed of a phosphate, sugar, and base. We have our covalent bonds on the backbone of DNA and our hydrogen bonds on the bases. I'm going to skip cell cycle. DNA replication happens during the cell cycle, right? It happens during S phase. And we've got several enzymes involved. Again, they're all these moving pieces. They're proteins, and they're enzymes, remember? And they all have a job to do, right? So we've got helicase. It's coming in here. It's unzipping the DNA. We have this DNA polymerase going toward helicase, right? Synthesizing 5 to 3' on the leading strand. The lagging strand's going in the opposite way because it also has to synthesize 5 to 3. And DNA is anti-parallel, so it can't go the same way. And then there's some other enzymes mentioned before. Topoisomerase kind of prevents the supercoiling that would happen here if you started to unwind it. And then ligase is that glue at the very end that helps kind of seal up any covalent bonds that haven't been made yet. Moving through to protein synthesis. And by the way, we're in unit 6, so I got to kind of keep going. Protein synthesis is gene expression. It's transcription. It's translation. So DNA to RNA, RNA to amino acids. Locations, went over this the other night as well, but pretty easy because in prokaryotic cells, they don't have a nucleus. So everything happens in the cytoplasm. And then the eukaryotic cells, remember you and I, our eukaryotic cells, we have nuclei and our DNA lives there. And so that's where transcription has to happen. Then the mRNA moves out to the cytoplasm to go to the ribosome. Looking here, if we zoomed in at transcription, we have RNA polymerase. It's moving in this direction, right? So it's looking at these nucleotides and it's going to give that complementary RNA nucleotide there. And I want to just talk about how does RNA polymerase know where to go, right? Like this is a gene, right? This gene is being transcribed, but like, how does this enzyme know where to go? So think about that. How does this enzyme know where to go? True. So the primer, good. So yes. Yes. Okay, so there's two things. So the primer is going to lay down some RNA nucleotides for the RNA polymerase to begin to attach new nucleotides to it. But really, it's about your promoter sequence. All right. So the promoter is like, I think of it like somebody who's hosting a big party and they're promoting it, right? They're like passing out flyers or they're probably posting it online. They're promoting it. They're like, hey, let's go, come to this party. Kind of like how we were promoting 5-able crayons, right? So the Tata box is an example of our promoter where it's a sequence of like TA, 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 TA. And that's, or another analogy, like if you've ever like flown anywhere or gone anywhere where someone's like in a parking lot, like at my church, we've got like a parking lot person who's like, come here, park here, you go here, right? You're promoting, you're directing, you're like, hey, this is where this is happening. So that's what your promoter sequence is. That's how RNA polymerase knows what to do. And then I want to talk about the coding versus non-coding strand, because some of you mentioned that the other day. And again, it's a tiny detail. And so if you don't get it tomorrow, it's okay, right? Tomorrow's the exam, whether you like it or not, it's happening. And you're going to do the best that you can do. But if you're feeling like, I just don't know anything, you know way more than you know. And getting a good night's sleep is the most important thing. All right. So this question about what's the difference between the coding and the non-coding strand, it's the strangest thing. But the coding strand is up here and the RNA polymerase doesn't touch it. And you'd think, like, why isn't RNA polymerase touching the coding strand? Here's why. When the RNA polymerase makes the complementary RNA transcript from the non-coding strand, that transcript is identical to the coding strand then, right? Because it's complementary to the non-coding strand, right? So it's identical. If you look here, I mean, besides T and U, obviously, but look here, G, G, C, C, A, C, C. So your RNA polymerase goes to the non-coding strand to produce the exact sequence, RNA sequence of the coding strand, okay? Crazy detail, not a big deal if you don't get it, okay? Okay, post-transcriptional modifications, mostly happening in eukaryotic cells like us. Introns go out, which I know sounds counterintuitive because it's in. You're like, why aren't they in? But exons are expressed, okay? So exon, 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 they are expressed, so they stay. Also, the word splice sounds like you're cutting things, which actually splicing means you're putting things together here. Kind of silly. Also, your RNA gets a poly A tail over here, and then it gets a GTP cap, which potentially has a role in like bringing it to the ribosome, but you don't even know that. But yes, RNA modifications that happen before translation occurs. In translation, similar sort of steps where you have initiation, because you're going to have, you know, a promoter initiation site, right? For AUG. It's not really a promoter, but it's AUG, the start codon. Then you have elongation, which is just the reading of the transcript, the tRNA bringing the amino acids. But I think something's missing here. So take a moment and look at this diagram. What's missing? Ooh, I like someone's comment that introns are intruders and they need to be cut out. All right, so what is this diagram missing? Termination. Yes, you were also good. And hey, if you ever need a break and we're just piling through, right, because there's only 40 minutes left, go ahead and walk away. Totally fine, right? Like if you miss something, it's not that bad, right? If you need a break, you need water, you need to stretch, or you need to go to sleep right now, go do it. Okay. Yes, but we are missing termination, and that's going to happen at that stop codon, right? There's going to be a codon that stops it, and then you've got your transcript. Now we have this primary structure, our amino acids transcript, and it's a protein now, but it needs to fold before it's actually functional. And so I'm going to go through this quickly as well. There's different types of bonding and interactions that happen to go from primary to secondary to tertiary, okay? The one that's including covalent bond is your primary structure. These amino acids are covalently bonded, which is really good because covalent bonds are strong and we don't want amino acids to be able to just spontaneously separate from each other, right? If the protein unfolds for a moment, they can come back together, that's great. It's not until we get to the tertiary level where we get interactions between the R groups. And if you remember, every amino acid is identical with the exception of that little R group off the central carbon, right? So some R groups are really simple, like there's a hydrogen, or there's a CH3, or there's something really big, right? And depending on that R group, they might be hydrophobic, they might be hydrophilic, and based on that, they might be acidic or basic, positive, negative, right? So it's not until that tertiary level that R groups start to come together and interact to give you that functioning protein. All right, I think we got one or two questions for practice. I think Rashi's going to come on and do these for you.
An mRNA transcript must be modified in order to survive outside of the nucleus. In addition to a GTP cap, a transcript receives A, exon tail, B, tRNA placeholder, C, poly A tail, or D, intron cap. And the poll is open, so go ahead and vote. Okay, there's 100% votes for C. Good job, guys. That is correct. And then a third concept check. A bacterium picks up a floating piece of DNA in its environment. This is called A, conjugation, B, transcription, C, transformation, or D, transduction. And the poll is open right now, so go ahead and vote. So the majority of people are voting for C, transformation, and that is correct. Good job.
Thank you, Rashi. All right, moving on to regulating gene expression. Also, side note, we have how many people here tonight? 300 and something, 300 and a wild amount, 300 people. So know that our student hosts and myself are really trying to get to your questions, but it's sometimes hard and or impossible. Okay, so don't think that we aren't trying, but everyone's trying really hard to get to your questions, but there's 300 people. So let's keep moving. Regulation of gene expression. Why do organisms need to regulate it? Why can't they just express whenever they want? Okay, Think back to the cell signaling unit, right? Unit 4, where there has to be some sort of messenger that potentially the message is to go ahead and express genes, right? Like as you are all growing up and, you know, your body changes based on hormones, right? And those are messengers that then tell, hey, you know what? Go ahead and increase bone density. Go ahead and increase muscle mass because this person is getting bigger. We have over 20,000 genes. We have 20,000 genes in every cell. If every cell was expressing every gene, it'd be a huge mess, right? So that's one reason to regulate gene expression, just one. There's lots of others, right? But just wanted to kind of ground you in that before we moved into the next part. I mentioned transcription factors the other night, and there seemed to be some confusion about it. So transcription factors are proteins that interact with enhancers and other sequences on the DNA, like the promoter, to help kind of organize the RNA polymerase. Certain genes need certain transcription factors or proteins, we'll call it proteins, right? They need certain proteins to be present in order for that gene to be expressed. So it's just another way to regulate on a different level. So we don't have to regulate in terms of, hey, we're going to supercoil the DNA and make it impossible to get to. But transcription factors can just kind of help organize everything and either say, yes, it's happening, or no, gene expression is not happening. Remember, so much of this test isn't necessarily about like all the things you've memorized. It's about, you know, they might give you a whole paragraph about this one type of transcription factor that does something you've never heard of before. But then the question is going to be based on what that thing does. And they've really told you all about it in the prompt. So I encourage you that when you see wild words you've never seen before tomorrow, you know, when they call something like TF1WAXG and it's a transcription factor, don't freak out, right? Just be like, all right, it's this thing. I don't need to know what it is. I just need to read the prompt and try my best. Okay. Prokaryotic regulation. The other night, I went over the lac operon. This is the trip operon. And operons, again, if it's one of those things where you're like, I'm just with operons, I can't. That's okay, right? Like it's the last night and you're here and you're trying your best. So, but what an operon is, it's basically a switch, a regulatory switch of, it's an easy on and off that can then regulate several genes that have related functions. So here, if I'm looking at this diagram, it looks like there's a gene over here that then makes a protein called the TRP repressor. And the TRP repressor interacts with this TRP, which is tryptophan. And when it does that, it changes shape. That's what I'm just looking at from the diagrams, not because I know it. I'm just saying, like, look, this looks like a different shape. It interacts with this other shape, and now it looks different. Because of that conformational change or shape change, it fits on the operator, okay? Which normally, now we've got this repression, because if this operator is blocked, this P is the promoter, which you don't have to memorize. They would give you a bunch of information in order to do this. But if that's blocked, then we can't have RNA polymerase come in. All right, so this is something that's going to be a repressible operon because it's not always repressed, right? It can be repressed when needed. Jumping through into mutations, explain what a mutation is and how it could happen, and then describe the potential impact of mutations. I'm going to just go through this because we're running out of time. Mutation is alteration in DNA. It can happen from external factors or occurring randomly, like in DNA replication. The impact of a mutation, the amount of protein could change, the type of protein produced could change, and then impacts could be anything: positive, negative, neutral. Generally, it's neutral. Could alter the phenotype, but generally it's neutral. Unit 7, we're going to do this in like 10 minutes, maybe, yeah, 12. We know Darwin, right? Came up or figured out natural selection. It's this mechanism for evolution. If you were to have to tell a story about an organism using natural selection as your guide, you would want your story to include that there was some sort of variation in population, right? Some sort of struggle to survive. Was there food? Was it a competition thing? Maybe an environmental pressure, right? Something changed environmentally. There was drought. there was something big that had big temperature change. And then the key is that certain phenotypes are more likely to survive and reproduce. Evolutionary fitness, you should know how it's measured. It's not about how well you can blend in. It's not about how easily you can get food. It's not about how well you can avoid predators, right? Those are all part of it, but ultimately, evolutionary fitness is measured by reproductive success, right? If all of those other things, the camouflage or behavior that helps you, whatever, if it doesn't lead to better reproductive success, then you are less evolutionarily fit. Okay, evolutionary fitness has to do with reproductive success. Are your genes being passed on to the next generation? Speciation, a new species, or what is a species? It's a group that can interbreed, produce viable, fertile offspring, which means viable just means alive. Are they alive, right? Fertile means they can also have offspring. And then it doesn't interbreed successfully with other populations. So how does this happen? Gene flow must stop. So if I were to have one population split into two somehow, and then all of a sudden gene flow stops, but how does gene flow stop? And a lot of you did really well with this the other day, so I'm going to go kind of fast. But if we have an original population and there's some sort of barrier, right? This is giving an example of a geographic barrier. That geographic barrier could eventually lead to reproductive isolation. And there's a bunch of different types of reproductive isolation, because why not? This is the college level class, and let's just keep hammering in the details, right? And then speciation could occur when all those things are in place. So if we think of examples of reproductive isolation, a lot of you came up with good ones a couple of days ago. Behavioral isolation, so like birds, right? Of all of a sudden these two separated populations, all of a sudden, not all of a sudden, over time, have varying like behavioral dances that they do to get a mate. It's kind of like it's kind of like how like your teachers or your parents don't quite understand your slang, right? Like I think every day I learn a new word that I didn't know the meaning of it until my high school students told me, and I was like, what? So it's like I've been separated from being a high schooler for 20 years, and I'm like, I have a totally different language, right? So anyway, there's also, I'm just going to go through these ones right here. So geographic isolation, sympatric speciation would be like that behavioral thing. They're kind of near each other, but then maybe one group starts to act a little different than the others. But then the reproductive isolation that comes from that could be, a new habitat. Temporal, which means just like time. Like maybe you're a fruit fly that feeds on one type of apple that only ripens in June, whereas you're another fruit fly from the same population that happens to prefer the apple that ripens in, September. Mechanical would be like actual fit, right, of like one structure to another in order for fertilization to happen. Post-zygotic barriers, that means the zygote was made, but then either the hybrid dies or cannot have babies, like a liger can't have their own babies. Evolutionary relationships, we look at trees for this. I guarantee you there'll be a tree tomorrow. If there's not, you can, yell at me on Twitter or something. But there's usually one, because it's really good and really important. And we can see who's most related. If they're closer together, we'd say they're more closely related. Way over here, right, it's going to be your common ancestor at that singular node. And then these two would be kind of the outgroup, right? They're out here off to the side. Okay, outgroup. These are kind of the more related species with each other. If we were to look at 3 methods that have used to investigate the phylogeny of organisms, we talked about fossils the other day. Anatomy and morphology are synonyms. Someone the other night was confused about the word morphology, but that just means like, think like I'm trying to think of like metamorphosis. Remember when you maybe learned that in elementary school when a caterpillar becomes a butterfly? So it's about structure, physical structure. Embryology, right? Like so many organisms look exactly the same in the beginning, and the same genes are actually being expressed. We ultimately love looking at molecular traits or biochemical. We like to look at DNA, proteins, amino acids. Those are legit. Okay, moving on. Oop, I just took someone's concept check, but let's just go fast. Phylogenetic trees show evolutionary relationships between organisms. We still have one more unit and we're almost done. Here we go, last unit. So then we will have time for practice. Ecology, Unit 8. It's study of the organisms and their environment. I had this picture of my son up here because he is outside and we've got like abiotic factors around him. We've got clouds and air. There's the temperature. And he's holding a snake. Right there's its little head. And he's outside, he's interacting with his environment, right? He's stepping on leaves and he's maybe stepping on ants and insects. He's picking up snakes that also have mice in their belly. And he's interacting with it, right? If we were to look at 2 abiotic factors, it was kind of what I listed, temperature, the weather, water. There's probably water because he's wearing his rain boots. Biotic factors, those would be the living things, right? So it'll be the worms under his feet. It would be the snake and the mouse. When you are studying ecology, there's a lot of different frames and a lot of different like zooming in or zooming out. So you can look at things from the population level, which would be like, hey, I have a population of elephants. Oops. And then the next level up would be community, because it'd be like, hey, we've got elephants and we've got lions. How are they interacting? Let's watch. Let's study. Then the next level is ecosystem. Okay. Ecosystem is going to be all the living things plus the non-living things. So population, community, ecosystem, biosphere. It's kind of like, if you think about it alphabetically, it's backwards from smallest to biggest. So P, C, E, B, like backwards. Hopefully that helps. Right. Behavior and communication, these kind of things are common sense in a way. It's like, oh, it's behaving this way or it's responding to its environment in that way. It's ultimately it's being favored by natural selection, that if you behave a certain way and communicate a certain way, if it helps you survive or reproduce, then it's going to maintain throughout generations. Okay, energy flow. So there's this real life story in the, oh my gosh, what national? Yellowstone. Yellowstone, which is like one of the oldest national parks ever. They were looking at how climate change affected the ecosystem. And this is just a draw up of like a very simple food web. And what they were looking at are these white bark pine trees, which are over 700 years old. They've been there forever. The bears rely on them. The squirrels rely on them. So many other organisms rely on them. But because of climate change, this beetle, who has always relied on the tree, the beetles are actually increasing in number. Okay, because the temperature is changing. So they're living longer. They're actually able to live in higher elevation because it's not as cold as it used to be. And so these beetles are skyrocketing and living longer. So the white pines are declining. Okay, so think about all the impacts there. So take a moment. You can say it in the chat. What are the impacts here? Beetles are increasing. White birch pines are decreasing. This would be a really good example of a question you could see on the test. They'd give you some story, some background about what's happening, and then you'd have to make sense of it, right? Predict what's the impact, what's going to happen, right? I see some people saying that squirrels will decline. Good. Clark's nutcracker, highly affected as well. Be really careful about words like it'll really affect or not affect. They'd want you to say it, like be confident in what's happening. Say, oh, the squirrels would decrease, right? The bear would have less of a food source there. Thankfully, the bears eat the grasses as well, right? So they're not going to completely starve. But yeah, you can, don't be so general. Don't be like, oh, it will change, right? Be specific. Say, oh, they would increase or they would decrease. It wouldn't necessarily cause a collapse of the ecosystem unless they gave you some really specific information about the keystone species is either removed or declining rapidly, then that could happen. But But yeah, just be careful about those big statements like, oh, everything will die, right? But be confident in what you do know, that like, hey, if these are going to go down, these trees, less of them, right? Or maybe they're less nutritious or something like that because of what the beetles are doing. But what I did read from that study is that in a 700-year-old tree, if the beetles swarm it and are doing well, it takes 2 days to kill the tree. So it's like actually killing these trees, which it didn't used to do because there weren't as many beetles. It was like a tiny bit, right? And now there's lots of beetles and they're living longer. So really crazy bad things. All right, I'm going to skip that. Population growth, some things that you'd have to know, right? That exponential means we've got no constraints. It's reproduced like crazy. There's no limit to population size. Carrying capacity, right, which is the letter K. There are limits, right? It's realistic. Most things are logistic growth, right? You might have a rapid thing like the wildebeests, right? They went from having like 100,000 to 1.3 million in just a couple of years out in the Serengeti. They hit their carrying capacity. And that doesn't mean that they like stay at 1.3 million perfectly, right? It's like the stock market. I said that the other night, where this straight line is really a zoom out of like, it goes up over the capacity, under the capacity, over the capacity, under, but relatively stable at that carrying capacity because of things that are limiting the growth. These are the equations you'd have to work with. I think we're going to do a quick diversity index problem, but any of these, really, it's just algebra. So use the letters that they gave you, right? Like N is population size. So start there. If they mention carrying capacity, then you better bet this is the equation you should use, right? If they give you R max and they give you N, this wild thing that maybe freaks you out, dN over dt, that's just telling you what the change is. What's the change in the number, right? dN over dt, almost think of that as like that's just change in the N over time. So you don't have to solve for dN, you don't have to solve for dE. That's just one quantity. We have 20 minutes left, everyone. You got this. We can do this. I'm tired too. It's 9:40 and it's late in Michigan. So if we think about biodiversity, what are some scenarios that would influence species diversity? Well, changes to your environment, natural disasters, tons of human impacts, right? If we come through and just clear out all one species of tree because we love it for our tables, we're affecting species diversity because then everything that lived on those trees as well has nowhere to live. So we've just wiped out like, a bunch of species. I mentioned the keystone species changes earlier. Big importance. Similar to genetic diversity, the more diversity you have, the more resilient the community is to changes. So the more diversity, the better. When you calculate it, I'm going to go back here. When you calculate diversity, the closer you are to 1, the more diverse you are. Okay. I just wanted to show you some real research because this is also just this kind of stuff comes up and you're like, I don't really know anything about this. I've never heard about it. Well, that's what they're testing, right? They want to see, can you look through a study and give me some information? So this is from Data Nuggets. I don't know if your teacher ever uses it, but I love it. It's out of Michigan State University, and they basically like summarize real research in a way that works for high school classrooms. There's this caterpillar that throws its poop. It's called frass. Frass is its poop. It throws its poop like 2 meters away. It's wild. And so that they're thinking that it's eliminating like this cue that might tell enemies that, hey, there's poop, which means there's probably a living thing nearby. So these caterpillars don't roam around like most do. They make shelters. And so if they didn't throw their poop, they would just be surrounded by their poop. And all these different wasps would be able to smell it and find them. It'd be like an easy target. It's like when you're playing hide and seek with a little kid and they're like, come find me. And you're right there. You're like, oh, you shouldn't have said, you know what I'm saying? So the caterpillar's not throwing their poop. Maybe something, maybe they'd get killed. So what would be a null hypothesis? If the hypothesis is that the poop ejection, the poop throwing behavior, eliminates cues that could signal the location of the larva or the caterpillar to potential enemies. Null hypothesis could be that frass ejection behavior does not signal larvae location, or just something more simple that there's no relationship between frass ejection behavior and predation. For your null hypothesis, you're just stating the opposite of what the hypothesis is, right? That there is going to be no relationship or there's no connection between the two things that the scientist is saying there could be. All right, so this is the real data from the study. And basically they measured the percent visits made by the wasps and the percent time on the shelter and the leaf. So two things they were measuring, which is your dependent variable, so it's measured. And the two things they're comparing is a shelter comparing beads, which is actually their control because it was just like a bead that didn't smell. It was actual plastic bead. It looked like same size as the frass, but doesn't smell like it. And then they had shelters containing frass. So they're comparing two buckets of data, right? They're not doing like a time thing. They're not doing a sequence of temperatures, right? They're comparing two separate things, two buckets. So think about what graph would you do? I already gave you the dependent variable, so that's what's going on the y-axis, but what would your x-axis look like here? Okay, hopefully you said bar graph, because when we're comparing two buckets of data like that, you bar them, okay? If you're doing something like your independent variable is a bunch of different temperatures, right? So it's like from 0 to 100, then you're gonna wanna do a line graph. And actually something that an AP reader told me was that if you are in doubt of what type of graph to do, you could do, instead of a bar graph and a line graph, you kind of do something in between. So you would like put a dot at your points and don't connect your points, but just put a dot at each of those data points. And then even from those dots, you could put your error bars. The last thing I want to talk about before we do practice problems is you might be asked to draw in error bars. You also might be asked to analyze error bars. If you took AP Stats, you're like, yes, I know this, I'm good. But if you didn't take AP Stats, this is another like low-hanging fruit that like, even if you've never learned this before, being exposed to it tonight will give you that extra edge tomorrow, okay? And so I just hope tomorrow when you're sitting in your exam, there'll be certain questions where you think, oh my gosh, yes, like we just talked about this exact same thing yesterday. And that's just going to give you a boost of confidence. And it's going to give you a boost of just excitement that you are doing well. Okay. So I just hope that happens. I just can't wait to find out. Anyway, we've got data. And if we look at mean means average, right? So the average percent visit on the control was 30%. The average for the actual poop shelter, right? They put poop near the shelter. Those were a lot of visits, almost 70%. If this was a freshman biology class, we would call it a day and be like, what? Look at that, 70% to 30%. Obviously, there's a significant difference between the two. However, in an upper level college class, we really dig into where that mean came from. Like, where did that average come from? Because you could have, an entire, like my kids go to a school that's kindergarten through 12th grade. And so if I said, yeah, the average height of this school down the street is 4'9", you'd be like, what? A school 4'9"? Like, what kind of school? What is happening? But if you dug into the data, you would see that all the numbers from the whole set deviate from the mean a lot, right? So if we looked at every number that came up with that mean of 30 or mean of 70 visits, we might be like, oh my gosh, like some of the visits made were like 80% at the control, but it's still average to 30. To counteract that, we do standard error of the mean, so SE or SEM, same thing. So here it's SE. I think on the test tomorrow, you might see SEM. It means standard error of the mean. It's even a part of your calculation sheet. But here are your standard error of the means. And they might say, hey, here's the standard error. Draw error bars to represent plus or minus 2 SE. You're like, what is 2 SE? 2 SE means you just double these numbers. So if I doubled 3.1, I've got 6.2. If I double 3.8, so on and so forth. So you double those numbers, and then, so here are my numbers doubled. And then you plot your error bars. And so I doubled those numbers, and it's plus or minus. And I know it might be hard to see, but if we go back here, for example, on the shelter with frass, it was 70-ish%. My plus or minus number would be plus, let's just round up to 70. So double standard error would be 6.2. I'm just going to call it 6. So the highest we go from 70 is 76, and we do, remember, plus or minus. So I would also subtract 6 from 70 and call it 64. So basically what it's saying is that within that data set, because of the standard error, most of my numbers, like 98% of my numbers, are between 64 and 76. Okay. And for this number, I already have it written here. In 95%, I think I said 98, sorry. 95% of this mean numbers, of the numbers contributing to that mean, are between 24.1 and 36.8. So basically you would go to where that mean is, that 30, and you would draw your line down to 24 and then up to 36. And that's where these lines are coming from. Okay. So it drew it up to whatever number it was and down to. So that's why it's plus or minus those numbers. Okay. In this case, our error bars are not overlapping, right? Like if I were to draw the actual bar, it's showing that it's statistically significant, that like clearly the shelters containing the frass were a huge signal to the wasps that something was living there. And that's exactly what the hypothesis was. If the error bars didn't overlap, you would say, hey, then what I'm seeing is not statistically significant. I can't say that I can reject the null, and so on and so forth. All right. So when they overlap, because like, look at this whisker bar, it's huge. Or sorry, whisker or error bars, it's huge, right? That means that like the data deviates from the mean a lot, which generally then is not a great thing. You want really teeny tiny whisker bars like this study had. They're really, really nice. And this is real data, which I love. Okay. Ooh, we've only got 9 minutes for practice problems. So I'm going to hop away and I'm going to let my student hosts finish the night.
Really quickly, could we go over chi-squared?
Yes,
a lot of people were asking for that. I think it's after the practice problems.
Yep, I will move forward to chi-square. And I think, was Jeffrey the one that was going to go over that? I think he had signed up to do it.
Yes.
Okay, so I will get to the chi-square. Here we go.
Okay, I guess I'll take it. So chi-squared, it's you take the this is like the main formula that you have to know. O minus E parentheses squared over E. That's basically what you're calculating for every single like cell or unit of data. And then what you're going to do is you see this sigma symbol, you just add them all up. And then because the standardized, I guess you can say, the standardized p-value for AP Bio is 0.05, you take that. And then your categories of data. So for example, you have, let's say you have an experiment where there are mice in the middle and like 2 tubes where you can go, then you would have 2 categories of data there because there's 2 like areas where the mice can go. So then you take those categories, that number of categories, and you calculate what's called the degrees of freedom. So your degrees of freedom is just the possible outcomes or the categories minus 1. You can see that in the little blue bottom right area. And then what you're gonna do is you're gonna basically take like XY coordinates. So let's say our degrees of freedom is 3, okay? And then we take the standard 0.05 p-value, and we discover that our chi-squared like comparison value is 7.82. So if our sum that we just calculated before, the entire O minus E squared over E thing, if that sum is bigger than the chi-squared table data, then what you're going to do is you're going to reject the null hypothesis. So the null hypothesis, at least my copy-paste one, is there is no significant difference between the observed and expected data. And because the entire sum surpassed the chi-square table data, then there was a significant difference, and therefore you have to fail to reject the null hypothesis. And then you cannot reject the null hypothesis. You have to fail to reject it. And then if it's lower, then you can accept the other hypothesis. That was an incredibly hasty explanation, but yeah, that's kind of just how it works.
Thanks, Jeffrey. I appreciate that. Yeah. And to, with chi-square, what I would do tomorrow morning, or not tomorrow morning, tomorrow afternoon, on your equation sheet, go to this table that they give you and give yourself a little key and draw an arrow to the right and say reject the null, and then get an arrow to your left to fail to reject the null. And it's just one of those things. If you can plop that out of your head and put that there just in case there's a chi-square. Again, there's no guarantee. There are some years where there's not a chi-square. There are some years where you have to do one and some years that you don't. So again, if it's one of those topics where you're like, I just can't get it by tonight, that's okay. Right? It's just a test. You'll be just fine. Okay, so we only have 4 minutes left. Let's see here. Rashi, do you want to come in and choose one question, or do we want to just do our final remarks?
I think we can just move on to the final remarks.
Okay.
If I know a lot of you guys were asking about like the Simpson Diversity Index or more practice FRQs, so those are all in the slides, which is in the Notion. So make sure you guys go and check those out if you have the time before your exam tomorrow. And then we can move on to the next slide. So exam details. The exam is Wednesday, May 10th at your local 12 p.m. time. Please show up on time at least 15 minutes before this exam begins so that you're not stressed out. You have plenty of time to sit down and take a moment just to relax. The other day, my sister was like, oh yeah, I think it was her AP Computer Science Principles exam. She was like, it begins at 1 p.m. It did not. It began at noon, and I'm really happy we double checked that because that would have been really bad. So please make sure you show up on time. The content AP Bio exam covers 8 units. Those have all been listed below, and it's a 3-hour exam. You do get a break in between, so bring a snack, bring a water bottle, and make sure you aren't sleep deprived.
Mm-hmm.
The exam layout. So if you didn't know already, there's 60 multiple choice, individual questions and question sets where it's like A and a B question, and 6 free response questions. 2 of them are long, 8 to 10 points each, and 4 short, 4 points each. You'll have 90 minutes for both sections. So again, make sure you bring a watch and allocate your time properly so that you're not spending too long on a specific question. And then we'll skip ahead. So time management, you have about 90 seconds per multiple choice question. Some of these might just be instantly 15 to 30 seconds to answer. Others might take you several minutes. So make sure you aren't wasting time. And for your FRQ, you'll have about 20 minutes for each long FRQ, 10 minutes for each short one, and 10 minutes to account just for reading the various passages. What to bring? This is the most important part. Make sure you have a calculator. It's a 4-function calculator, scientific or graphic, that cannot connect to the internet. Please review the AP College Board allowed calculators. Bring a pen, a blue or black ballpoint pen. Gel pens are not recommended, and a pencil for the multiple choice section. And bring extras. Make sure your calculator is charged. Bring any extra batteries if you need to, and especially bring extra pencils that have been sharpened properly. A tip, please use your best handwriting skills tomorrow. I know you guys will be in a rush, but if your reader cannot read your answers, they can't score them. So handwriting quality comes before getting all the answers in this case. And some final remarks for the last minute. We are so proud of you guys for showing up for the last 5 days, 3 hours each day. We've all accumulatively been studying for 15 hours, which is insane. And I know you guys have been putting in a lot more effort on your own. You guys will be able to crush this exam tomorrow. I have full faith in you. I've seen the way you guys answer each other's questions in the chat, immediately jumping in to help people or solidifying the concept if they don't understand it, which is so nice to see. Thank you all so much for being so helpful to each other. But please make sure you sleep properly tonight and have a healthy breakfast tomorrow. Your mental health comes first, and no matter what score you receive on your AP exam, we are so proud of you for putting in the hard work and the determination to show up here every day and study with us. Please make sure to also take the AP Bio Night 5 survey. It's linked in the green chat box, and Jeffrey also drops it in the chat. And thank you guys so much for joining and for doing this AP Bio cram with us. And as always, follow @ThinkFiveable on social media.
Thank you so much, Rashi. Everybody, we don't even have time for questions, but you're good. Go to bed. Eat something good in the morning. And we just thank you all so much for your participation and for your effort and your positivity. You all just were so great. I really enjoyed it. And just best of luck tomorrow. You're gonna do right. All right, good luck. See ya.